and Ivan. So today we will have a way of image analysis. So Hesti graduated in mathematics and obtained her master in statistical and computational data processing in Madrid. So Hesti finished her PhD in 2021 in mathematical engineering, also in Madrid. So she will be here also with Ivan, which is a software engineer. So they are both working at the Optical Cell Biology Lab at IGC. So Hesti is an expert in biomedical image analysis, and she's currently a postdoc researcher. And her research focuses on contributing to biological discoveries by exploiting image processing techniques.
Thank you very much. You're welcome. Okay, so a lot of math around, but we won't see that much math today. I think it's always going to be in the background. So before starting, I heard that you were very excited about doing image analysis, but I would like to know why, because I heard different stories. So it would be nice to listen from you. When do you think you can use image analysis? Or why do you think it is important? Or why do you think you should have this skill? Any volunteer? Who has already some data to analyze? It's very early in the morning. Okay. And what softwares have you used? Pitchy? Anything else? Uke? Nothing else? Imaris?
Yeah,
Okay, okay. Who has written sometime a script to automatically analyze at least 2 images? Yeah? Where? In Fiji, in imaging macro. Okay. And what kind of image analysis have you done?
Identification of fluorescence.
For fluorescence? I mean, how do you do that?
That's much on the expert level. I'm just interested to see how many cells are expressing what we are interested in. So it's really just counting stuff that are better fluorescence. But I would like to improve it for example, quantifying the level of expressions of the intensity of the fluorescence. That's
the intensity of the fluorescence. Okay, but that's data analysis, right? That's when you have your data from your images, and then very good. So then we start to talk about something else. Okay, so now you spoke about quantification of fluorescence, but where do you think you can use image analysis? For what do you want to know it?
That's a biological question.
Okay, to answer, but sometimes you can use it to quantify how many cells you have in your field of view, right? What else can you do with that? Measure the cells, the size, the morphology. What else? Colocalization. Is anyone here doing light cell imaging? And also tracking, for example, or shape changes. What else besides answering your biological questions? What about quality controls? Right? So you saw something about expansion yesterday. Why do you want to do expansion microscopy? I mean, you expand your samples, you have better resolution, and then you have a nice image figure, right? And that's it. Okay, so usually image analysis is not only for your cells, it's usually embedded in whatever you do in microscopy. It can be quality control of the experiment you did, and you may be asked to prove that you did the experiment correctly, and for that you need to compare conditions. It might be because you want to do some analysis of the super resolution, or then doing some biological discoveries. And to give you a bit of glimpse of what we do in the lab and where do we use image analysis besides shape morphological analysis, that I think we don't do any of that. Of course, we can do expansion microscopy. Probably Mario showed you this gist yesterday. So then here what you have is the organoids, and then you can build atlas across different gists, and then you can compare, right? If I treat might give you some kind of drug, then it can change the shape of the usual normal mean shape that I calculated by analyzing, I don't know, 100 kind of G cells. Of course, it can count the number of cells it has in this zebrafish. I don't know if he showed you this expanded zebrafish embryo. We can, of course, run into super resolution, but I guess Hannah also showed you some surf, eSurf, and so on. We can use it to automatize the microscopy acquisition. We can use to super resolve images, and many things involved in image analysis. So today, my idea is not to make you the best ones at programming a macro. I want to give you the knowledge of what are the things you can do and what are the technical considerations that you should take when you do image analysis, and where to go to ask for help. Because it's a whole field, it's super complicated, and each of you will probably need a different tool, a different method, and it's very difficult to cover all of it, okay? So the slides, I will share all of them with you. Also the exercises, the data, the emails are there, so if you want to shout for help at any time in the future, you can just drop an email and ask for it. So yeah, bioimage analysis. So when we do research and in our life in general, when we try to identify something that is different, what we do is visually compare across types, right? And identify patterns that tell us that something is going on, right? And this is very nice. We have a very impressive capacity in our brain and in our eyes to identify patterns when we are used to see some things, right? I can see, for example, if I have fishes, right, I can somehow say like, okay, this fish looks different from this other one, but I don't know very well why, but very quickly we are trained to identify patterns with our eyes. So this is something we've done for edges, and there's something besides visual capacity, and that motivates computer vision in general. With biomedical images, that's something that is super cool. That is, we don't only get information about what's going on, but we also get information about when, about how many, about with what intensity. So in general, the signals that we acquire with a microscope, with biomedical devices, are much more complex than just what we see with our eyes in reality. And that's why this is so important, because this is the data you do to do some research, right? But then the question is, can we trust our eyes? And probably you are very, you know very well this game. So A and B, do they have the same gray color or not? Who thinks they look the same? Who thinks they look different? Okay, so they are the same, and that's the problem, that our brain gets adapted to conditions. So if you enter now in a dark room, you won't see anything. If you spend 5 minutes, you will see probably something, right? And so our brain does these kind of tricks. Then what about shape? Are they the same? Do they look the same? No, right? But they are the same. And then here, if I ask you which is the avocado that is in the middle? Yeah, which avocado is center? Who thinks it's the first one?
I think it's the first and the third.
No, it's not the second.
There's only one correct answer. Who thinks it's the first? The second? The third? Yeah, okay. So that is very good. But now we're going to microscopy. How many cells are here? 7, 8. What? It starts, yeah, having some problems. Here, here, and there, and so on and so on. So, well, that's why we really need this kind of programming. That's why we need to have ways to count the same number of cells today, tomorrow, and when you are defending your thesis in 5 years, 3, or whatever, right? So then how do we treat these images? So images are basically a grid of pixels that have values, right? And these values are displayed in our computer. So in colorful images, what we have is 3 values, red, green, and blue. And then the computer, according to this combination of these 3 colors, will display yellow, or we display red, or we display green, and so on. And then we have the grayscale images, the one channel image that is a very basic acquisition that we do with a device that has, again, some values here. So this is the normal definition of an image, but then what is a biomedical image? It's this grid of pixels and something else. And what is this something else?
Coordinates?
No. What else? What is a biomedical image? What is the file that you get in the microscope?
The image starts
If I give you this set of pixels, and I tell you, can you please tell me what's the size of the object you are seeing? Can you tell me that? Scale factor. What is the scale factor? Of what? The scale is 0.5. Is that telling you anything? No. So what is a biomedical image? What else do you need? What do you need?
Metadata.
The metadata. So you need to know what is the real size of a pixel. Is it 3 microns? Is it 2 nanometers? Is it 1 centimeter? You need to know the wavelength you use to acquire the image, because you need to know what is the fluorescence you are getting. In medical scans, you also need to know some kind of radiation, just to understand whether the signal you are getting from the gamma, the X-ray, it's displaying water or not, because then we need to reconstruct to see actually the final scan you see, and many other things. And this is called metadata. And this is what is encoded in all those files you get in the microscope from Zeiss, from Nikon, and so on. And there's something very great that is called bioformats that you should really, get in your brain for all your life if you're going to be researchers working with images. That is what defines, so it's like a consortium that is able to read different image files from different microscopes across devices, across computers, right, to really make sure that this metadata is stored. So if you are collaborating, if you're analyzing your data, make sure that you never lose this file, because otherwise any quantification you do, it will be completely unreliable. Okay, nobody should trust it without the metadata. So then images. So we have I told you the very basic kind of images, a grayscale image, but then these numbers are not numbers from 0 to 1, right? We usually have bytes, and this means how much space do we give to the computer or to these pixels, or to the memory, to allocate different scales of grays. And then you will say, well, I usually want all of them. All of them is a lot of memory, but I'm pretty sure you are very used to see these 8 bytes images, 16 bytes, and so on. So then this is of course important for quantification, but again, here there's something that is kind of funny. So the computer can display many grayscale values, but how many can we see actually with our eyes? Eight? No. You see, so we see around 900 kind of, we are able to distinguish the contrast between 900 kind of gray values. I think some people more than others, so probably people in Greenland and so on, they are more sensible to whites and those things, and they see more than probably us. And then when we use these monitors, usually they don't display them. So that's why radiologists, usually they have very expensive monitors to display all these kind of grayscale values to really check the differences between tumors and lines and so on, okay? And then why is this important? Well, if you have this fluorescence image, the amount of gray values you have in the image is giving you information, right? It's the fluorescence value. And if we have very few, then we are not able to display all this range of values. And sometimes we need to have really many numbers to get the finest details in our images, right? And this is related to the contrast. And here, for example, I'm giving you 3 images, and they have different kind of bit depths and different kind of contrast. So here you see the original CT scan with all its pixels values. So usually here around the gray is the skull and so on. But if we don't use these gray values properly, if we modify with Fiji, for example, you convert your fluorescence image from 16 bytes to 8 bits and so on, then you can destroy part of the contrast, and this will affect directly to your quantification if you want to do segmentation. Or you can, and depending, of course, on how you modify it, you may lose information and so on and so on. And this is grayscale, but usually you see colors, right, when you use fluorescence. Why do you see colors? Are those images colorful? No. So then? Exactly. So we attribute lookup table, and this is what we do. So here, usually we display the values saying that 0 is black, the maximum, this is an 8-bit image, is white. But of course, we can decide to change it, and these are the lookup tables. So you see colors in microscopic images, but they don't have color. This is something virtual that we assign that is important for the beautiness, for the contrast, but also if we want to quantify, for example, for the segmentations, usually we show it with colors because we want to create contrast in a way that is easier to evaluate what we have in the image. And then this is something very important that I would like to highlight, because you are working with microscopy, and its resolution, and how resolution affects to whatever we are analyzing, right? So if here we don't see the same amount of information as here, and so on and so on. So what is resolution? There are different ways of defining it, but what is for you resolution?
Minimum distance that we can. So
you have class more than one day. So now, what is for you resolution? The minimum distance.
We can resolve it in 2 points.
2 pixels is the minimum distance between 2 points. What is resolution? Is it the size of an object? Is it related to the size of an object? What is the difference between a super resolution image and a wide field image? You distinguish better the details in the image. So resolution is given by the details we are able to distinguish in an image. And it doesn't necessarily need to be related with pixels only. So here we have the same guy, and the same guy or animal, I don't know, happy cell. And this guy and this one, they have the same size. The resolution of this image is higher because I can see more details in the image. And I can see more details first because I have more pixels to distinguish between the details, to allocate gray values, right? And paint things. So here I have only 6 pixels for the nuclei, or whatever is this. Well, here I probably have 100. So then I have more space, I have more pixels to allocate detail. If I upsample this image to an image of 1000 times 1000 pixels, will I have higher resolution? Right. I don't know if I'm showing it. So if here instead of 512, I get an image with many more pixels, but we see the same, do I have a higher resolution?
It depends, because we can only distinguish 900 shades, so to a point it won't make a difference for that.
The same image, I upsample myself. I just double the amount of pixels, but it's the same image. Is the resolution higher?
Should be in the definition.
I have a larger pixel size, but the resolution is the same because I came from the same image. So resolution is the details you can see in the image, is not the number of pixels, okay? And it will condition the number of pixels, of course, because you need a space to allocate the details, but the resolution is usually given by your microscope and not by the number of pixels. And this is something very, very, very important. And it's related to the metadata, and it's related to the objectives you use, and so on and so on, okay? So then here I'm showing you, this is a very nice figure from a perspective from this year. So what they did is to acquire same field of use with different objectives, right? They go from 20x to 100x, and they measure the roundness and the size of this exact nuclei, right? So the area, it changes a bit, right? According to the number of pixels, we have to allocate information of the nuclei, but it's not that much. While here, the contrast of this small mitochondria, it will be very different, depending on the objective we use. So this image, it has better resolution than this one. It has higher resolution. Probably the number of pixels is also higher. But if we want to quantify the nuclei, the roundness, the circularity, the area, it's most of the time the same, and no matter the objective we use. So this game between resolution, objectives, and the quantification you want to do, you need to always take into consideration before acquiring your images, right? So do I want to image some nuclei that are already quite big? Yes, then I don't need to go into images which have like a 0.5 nanometers of pixel size. On the contrary, if I really want to distinguish this contrast in the mitochondria, then I need to have a higher resolution. Okay, is this more or less clear? Yeah? Okay.
Sorry,
Yes.
I have just a question. I was wondering, someone mentioned definition.
Yeah.
How do we differ resolution from definition? Is it the same thing, kinda?
So resolution is something we use because we have the pixel size, like I told you, 0.5 nanometer for pixel size, and we are able to distinguish details that are the minimum size we are able to distinguish is 300 nanometers, for example. Definition is something we don't quantify. It's a concept that comes from artistic image analysis, and it's how do you say nítido? It's how defined an image is, but it's more related to the images you take in the phone. So define it means that you have a good resolution in your image and you're able to distinguish the details. So you don't have noise, for example, besides the very small pixel size. The definition is not a concept, is not a technical concept.
Yeah.
Because it's more visual. It's like, okay, I can see this image is very well defined. And you will see there's an exercise at the end of this morning that you will see how to measure resolution, but definition is not it's something you shouldn't use technically in a paper, for example. Okay. Okay, so what can we do with digital images? And now let's go. So we do image processing. And image processing is basically when the input of whatever algorithm we are going to use is an image, and we produce some sort of mathematical output. And this can be as creative as you want, okay? So it can be that the output is just location, if we do tracking around time. It can be that we just classify pixels between background and foreground for segmentation. It can be that we create a larger images, so we do super resolution. This is a storm image. Or it can be just we give a number, whether this field of view belongs to infected cells or not, okay? So it's basically an algorithm, an equation, if you want to think about it, where the input is an image, and an image can be a video, it can be a volume, it can be whatever you acquire with an imaging device, and the output is some mathematical element with some sort of image. And the way we use it is called bioimage analysis. And this is the part what I wanted to highlight. So image processing is just the fact of producing an output. Image analysis is actually doing your data analysis. It can be some statistics after you process your image. It can be some classification, and it's basically the process you follow to create knowledge with your data. And you can combine it in many different ways, but there's something there's something that you should consider. Whatever you want to do, there's always a tool specific for it. Okay, and somehow if you are really wanting to do this, you need to master your tools. You need to know very well that you can use Fiji, but maybe you can use 9 for your statistics, or maybe you should move to Elastic, or maybe you should use QPAT. Okay, and you should be very open-minded to learn new softwares. So then, because we like to have things a little bit organized, when we speak about image processing, we speak about tasks. Okay, so are we going to do a segmentation? Are we going to do tracking, registration, and so on? So the most common one is segmentation, because usually you want to identify your cells or your objects and then do some quantification with them. But we can also do just the detection, but not the segmentation. We are not interested maybe about the morphology, tracking, registration, denoising. So here is where you say this image looks more defined than this one, but what we did was to increase the resolution and remove the noise. Super resolution, and there are more, but these are the very basic ones. And today I will dig a little bit more into this one, because it's the most common one, and I think is the most useful one generally. Well, we will also see some others, like tracking and registration. And I think this is something that I like to highlight. I told you that you need to think about what you're going to quantify before the acquisition. And the reason is because depending on what microscope you use, how you prepare your sample, your images will look in one way or the other one, right? It's not the same doing electron microscopy than doing wide field. And this will directly impact on the analysis you are going to do afterwards, right? Not only on whether you can segment the structures, but even whether your data is going to be 1 terabyte or just 200 megabytes. Okay, and this is something you should consider, and you should know very well what technology you have accessible for you. And then the processing part is not just one segmentation. So sometimes, or I would say most of the time, we just don't do one of the tasks that I showed you before. We go through all these steps of, okay, maybe before segmentation, I need to clean my images, I need to do denoising, I need to super resolve them, I need to maybe identify objects and then segment them, or identify the objects, then track them, and so on. So there are a bunch of many different steps that you may follow in your research project. And for each of them, there's a huge bunch of open source tools, okay? And these, again, I would like to highlight, because these are, all of them, they are ready to be used for your analysis. Some are very specific for organoids, for neurons, for whatever. And there's this forum, the M.Sc forum, that you can always go there to ask for help if you don't know how to analyze your data. The bioimage analysis community is super active, and there's always someone there able to answer your questions. So don't hesitate to go there to ask. And then, of course, here is what I told you. I would recommend to think about these steps even before the acquisition, but also while you're doing the acquisitions and while you are getting some data, so you can really optimize the way you analyze it. And then there's the data analysis, and all this is what we call microscopy imaging in reality. Okay, and you should consider each of these steps when doing your research. So in other words, and this is from a comic from Pete Branke, the developer of QFAT, that I really love it. So forget about being in this place, comfortable space for you in science, because you really need to get some way to communicate with people developing algorithms and getting some very basic but technical knowledge to make the most of your data. So with this, let's go very quickly with segmentation. So segmentation is basically we take the image and we classify the pixels with labels. So we create segments in a way that is easier for us to quantify them. And this will allow us to count cells, so how many different levels I have, to analyze the morphology, or to differentiate between different anatomical regions of the brain, or many other things. Okay, so this is why it's interesting to do segmentation. And basically what we do, we have the image with intensity values, and we need to determine which ones belong to one group, like the group cells, and which ones belong to the group background, okay? And this we do it by assigning values, 1 or 0. So how do we do this? Well, in this case, it's kind of easy, right? The whitish kind of area, the light is 0, the dark is 1, but usually images, they are not like that. So we usually need to clean the images or extract features. So one very basic step or strategy to do segmentation is usually we filter our images. So we remove details because we want to segment big structures. So here I'm just filtering. Do you know what are filters? So not mathematically, but basically, you know that you can take an image and apply as something that is called filter, that you will see what is today, and the noise or enhance some features or whatever. So we can denoise as much as we want. So then we create a very big contrast into between the black. So all this is black, is 0, and the green area, and we segment. So a filter, and this is the mathematical part, this is an image, is a convolutional operation. Okay, so what we do is, okay, we take, for example, this is my image, and then for each pixel, what I will do is I will take the numbers around this value, and I will compute, for example, the mean. And this is the mean filter. And what I do is to repeat it for all the elements in my image, and then I get a filtered image, right? I remove a bit the contrast between these pixels, and I average all their values. So this, of course, operation, it doesn't always need to be a mean. It can be just a multiplication of each of these numbers by something that is predefined. And this predefined element, this matrix, this filter is called kernel. Okay, and depending on what is the operation that I do for the pixels, I will enhance some features or others. Is it more or less clear? Yeah? Okay. So this is called spatial filtering, and it's super useful to remove noise, especially. Okay, and this has been used for years and is still used for many things. So depending on whether you use the mean that I showed you at the beginning or the median, you will remove outliers or you will smooth your image. Like you have ever used Photoshop or anything like that? Instagram filters. Right, those that they make you like a super smooth skin, right? It's a medium filter, okay? So I think you all know about filters more than what you think. So then the other thing is, okay, we can clean the images, we can threshold them, but we can also do mathematical operations, because at the end what we have is just a matrix, right, with numbers. So imagine this is an image where the background is it's black and is 0. This is the other way around, sorry. And this is 1. So if we sum these two images, then we have 2 in the middle, 1 where there's only the circle, and 0 in the background. And we can do this with images in many different ways. Now I can add still a triangle, and then I will have 3, 2, 1, 0. So if I have this, what could be the result of this operation? What will I have in the middle? Yeah. What value? Here in the middle, what's the value in that pixel? What's the value here in this one? What's the value of this white shape? 2, no? Who doesn't think it's 2? It's 2, no? Is this one 2? So this is the other way around, sorry. So this should be white, this should be black. So this is 2. What's the value of this one? All right. 1. So 2 minus 1? 1. Okay, good. I said there were not going to be math, but okay. So what about here? What would I have here in this area? What's the value here? And here. So then here in the result. So okay, yeah, I guess this it's getting complicated. So basically, what we do is this with the images. So if I know that I have this shape and I want to enhance the triangle shape, I can subtract them, and this happens with images as well. If I know I have some noise, some fluorescent signal in my image that kind of blurs it, I can kind of estimate it with some filter. So I remove the details, and what is relying is the fluorescence. So if I remove it here, I will enhance each of the components in my image. Now it makes it's interesting, right? Okay. So this is super important. I'm telling it to you now. You will forget it in a minute. And this can solve many, many issues of your data analysis. Don't forget it.
So,
yeah.
Sorry, just a question. So is subtracting background the same, is a filtering process?
No, subtracting is an operation. I just subtract. A filter is when you compute for each pixel a value. So this you cannot get it by subtracting two images. You need to really especially do some math. So a filter is an operation that allows you to identify what is in the network of each pixel and then subtract some kind of features. Okay. Yeah.
And the last so the middle image is created based on the original image. Is that it?
Yeah. So we have this one. What I did is to filter it out, each of the channels, with a median filter. So then I don't have contrast between the spots, but I have the average fluorescence. And then when I remove it, I create contrast in my spots.
Can you lose data on that?
Yeah, of course you lose data, but you don't do this to. So yeah, when you subtract, whenever you do an operation, you are modifying the values of your image, and that is actually a very good question. So if what you want is to count how many objects you have here, you don't care, because you are doing this to enhance the information that you have in the image and say, okay, I have, let's say, I have no idea, but I'm assuming these are some sort of mitochondria. I'm sorry, I really don't know. So then maybe I want to measure the amount of mitochondria, like the density of mitochondria I have around the nuclei compared to the edges of the cell. So then, yeah, of course, you lost information, but what you are going to do is counting. If what you want to do is see whether these mitochondria are brighter than the mitochondria in another image, then what I would do and I would recommend is you identify where they are, you go back here to the original image, and then you quantify it in the original image. Okay? So then, yeah, of course, the intensity values, you need to be very careful by modifying them. Okay? Yeah. So we can still go with the spatial filtering. So of course, the filters we use, they can be larger, they can be smaller. And one very common way to use the spatial filtering is to identify whether there is any illumination pattern in the image. So if I use the filtering that smoothes incredibly my image, I would get this kind of shape, because actually if you see it from the background, this area here is brighter than the area in the edges, right? So then what you can do is, you calculate it just with a Gaussian filter, and if you divide the image or you subtract it, what you do is you clean this artifact and you have an homogeneous image. And why is this important? Well, because if you try to threshold the image like here, your result is going to be probably very crappy because the intensity values here are not the same ones as here. And when you use this rule of saying whatever is smaller than 0.5, I remove it, is not the same distribution here of values than what you have here. While if you homogenize your information in the image, and then you try to threshold, the threshold that you applied in the middle will also work in the edges. So of course, there are many ways of extracting features. I mean, this is what I show you is very basic, but then you will see Laplacian filters, gradient filters, Kubahara, linear, whatever differential filter, and so on. If you go to Fiji, I mean, there are many of them. And each of them will enhance the details in a way. And then the question is, how do we know what's the best way, right, to get these filters? Do you know the answer? Who wanted to learn deep learning? I expect one hand because a little bird told me that someone here said wanted to learn deep learning the first day. Who doesn't want to learn deep learning? Okay, so all of them, right? I mean, it's a black and white thing. Why? Why do you want to use deep learning?
Because we are looking and saving some work.
Yeah.
Save some work. That is the worst answer that you could do.
That's why there's need to deep learning. What is deep learning?
I'm gonna explain it, but so you didn't.
What is deep learning?
Huh?
What is deep learning?
So deep learning is a way to optimize. It's deep learning. Yeah, it's very difficult. So when we analyze data, we can define rules and filters, as I show it here to you now, and we can mathematically write equations that we want to apply to the data. Or we can leave the parameters of these equations open, and then what we use is optimization to know what are the best parameters, and that is called machine learning. So when these equations and these parameters belong to a very big set of data, like we don't have 2 parameters, but we have thousands of them, we are speaking about deep learning. So it's a set of algorithms that we need to train for them to know what's the best way to process data. And that is a very mathematical way of explaining, but I think it works much better. Okay, we will go into that. But the thing is that what we do with deep learning is actually, or with machine learning, learn what are the filters we need to apply. Because usually we don't know how to define them. So spoiler alert, because this is for the afternoon. Yeah? No? Okay. So basically I show you thresholding, but of course I think it would be very naive to say that we don't need deep learning to segment data, because it's what we use nowadays. And then what is important is that we don't start thresholding, but we start extracting features of our data to know how to segment it, to know whether here, if I try to put all this in white, but then I need to count these cells, probably is impossible because they are touching each other. So then I need to find a way, or in a smart mathematical way of saying, okay, here I have my edges. So then I can split the cells and I can count them, okay? And this is some feature that we can learn. Then, of course, we can start saying, okay, I don't want to segment the cells. I want to segment the boundaries, because that is going to allow me to split, to separate cells one from each other. And of course, we can do this many times without using deep learning, but I would say 90% of the time you will see deep learning, okay? So that's why I go into this. The same here. So it makes no sense trying to segment this image like this, because then if you want to split these different ovules of this, this is another Arabidopsis oval, it would be impossible. So what you try is to identify where the boundaries are, And then there are more things you need to think about it when you do this segmentation. So let's say you want to segment these nuclei here. So these are very easy to threshold. But again, they are touching each other, they are overlapping. So trying to get a segmentation that works here doesn't work very well. One thing some people do is they say, okay, I will just create a bounding box around each of my objects, and then I will segment each of the bounding box. And this works very well in computer vision sometimes, but it doesn't work well in biology because our shapes, they don't always enter in one bounding box, and we may have some that overlap one with the other one. So then I'm pretty sure some of you already heard about this Stardist algorithm. If you do nuclei, no one? Yeah, okay. So it's a smart way of saying, and this is why it's important to understand when we do image analysis that we can extract any feature we want. It's not just a thing of black and white. So what they propose here is, okay, I don't need to segment the objects. What I can do is try to calculate where are the centroid of my nuclei, some kind of boundary descriptive kind of feature, and then I just do some math to reconstruct each of the circles, right? And then in CellPose, that is the one we are going to use today at the end, again, is the same. If I try to threshold, I'm going to lose most of the protrusions here, right? Because they have a weaker sinus. So then what I try to do is, rather than going directly into the segmentation, I will try to define some features. That is a flow that is basically what's the distance from here to the center, more or less, for you to understand. And then I reconstruct the shapes and I have the segmentation. So segmentation is not only a black and white thing. You need to be a little bit creative. And the way that works for me, at least, is when I look at the images, what are the features that let me distinguishing between objects? So those are the ones that I should try to enhance, that I should try to put in my network to learn in order to get a segmentation. And there are many things on, so you don't need to worry, you don't need to codify them, but this is important for you to know. And then the second thing I would like to highlight is that whenever you do an image analysis task, you need to evaluate your results. And the first thing you have to do is to visualize your output. Never, ever process your images without visualizing what you get out of it, because even for me, if I do that, I may have some mistakes. So visually, we are very good at identifying whether things work or not, or whether it's what we expected or not. Of course, there are many different accuracy metrics, like the number of true positives, false positives, how much do the segmentation and what's the ground truth overlap between each other, what are the distances to the boundary, and so on and so on. And then one that is very important, that is how biologically relevant is the result I have. So imagine this is a mitochondria that you want to segment, and you can always do a circle around it. And this circle overlap like the entire mitochondria, so the accuracy, it's like 0.8. It's quite good. When I ask you, well, if you want to identify the length of this mitochondria, the circle is not a good chance. If you want to identify what's the diameter, is not either a good chance. So those biological kind of metrics that you are the ones who should define it and not the people doing the algorithm are as important as the accuracy. Okay, and you should always report them in your papers. So this is a very, very nice book. You can go into all the sort of metrics for segmentation. It has 100 pages. So if you don't know what to start with, this is a very nice guide. And let's go for the practical session in 50 minutes. Right? Unless you have questions. Any question here? No? Okay. Yes.
How much computing power do you need to run those programs that you mentioned?
Which ones? Stardist and CellPose?
I'm not sure. Like, can you usually run them on like a regular, let's say, computer?
Okay. So if you want to analyze your data in TG and you don't want to use deep learning, you can run it in the computer.
Deep learning, you always need a
no. So today we will use CellPose without a GPU. So for inference, there are many models start to use CellPose and so on that you can use it in your laptops without the GPU. But if you need to retrain them, you need a GPT. For that, there are two choices. One is what we are going to use today, is Google Colab. So it will give you around half an hour of computing power. It's not the best, but if you just need to do one image processing for your thesis, then it's not that bad, I think. Then the other chance is, well, either you buy the GPU and you say that, which I think it's not a very good choice. So either you try to find collaborators who have the GPU, or the institutes, usually they have, I don't know here, but usually institutes, they have some sort of bioinformatics facility that are very misused, I would say. Most of the time, I mean, they are super happy for you to go there and say, hey, I want to use the computing power. And you can use it there. But it really depends. So if you have, for example, electron microscopy images of the whole embryo, you can upload that in your laptop, even, but not even for visualization. If you have just 100 images of nuclei, you can do it in your laptop. So it's a big, yeah. Okay, so this is what we will do today. I think it's a bit ambitious, but I think we will manage. So yeah, let's go for the break.
So who has never used GitHub? No one. Okay. But do you know what it is? Yeah? No? No. No. Okay. So GitHub is a web page that uses Git. That is an application to keep track of the code we write. Okay, and it's a place where we can have repositories. Are you listening? No. Yeah. Okay. So it's a place where we store our code. We store our software developments. I store there my papers, so I keep track of what I write and what the last author of the paper changes in the paper, just in case I don't like it. Feedback to what I liked. And the reason why I think it's important for you to know it is because now with the AI era, more and more developments, even if they are user-friendly, they are stored here. And the wikis, the documentation, they are in our repositories as well. We will see it later. But also, it's an amazing place for you to ask for help when things are not working. And here, same as the forum, that I think is really, really the best place to go. If you are trying to use, for example, a Fiji plugin that has a GitHub repo, here I'm showing you one of the statistics that I worked some time ago. You can always go, you're not seeing. No? No?
So if you go to the home in your computers, you will find the FijiM folder. And if you open that, you should have Fiji inside the executable file.
But we need to open it now?
Yes.
Okay. Alright, we didn't know where I was. So,
Okay.
this is the GitHub repository, okay? Just one, which has some sort of statistics, right? And here, in all the reports, you can always go to issues, and here create a new issue with a new question. Hey, I tried to run your code on this type of images, it's not working, my images are black. I have this error, and you just copy paste the entire error. And developers here, when we write those, we get directly an email through our mailbox saying somebody is trying to use your code, which is something super good for us. So we are really, really happy. And somebody wants to really use your code, so they open the issue, which makes it even more exciting, because then they're going to cite your work, and they don't manage to make it work, or you have a bug. in your code. So this is really the way for us to know when things are working or not, and for you to ask for help. And this is empty, but maybe I can show you one from zero cost that we are gonna use it later. So here I have 42 open, 145 closed, and I can just open any of these. All the people who open these issues, they are biologists. And Ivan and I, we are actively answering them. And they, oh, here maybe this one is not the best one, but there was one here. Okay, here there was someone who was trying to use the segmentation model. It was not working. They were putting plots of the data, what were the problems they were having, why it was not working. I was actively trying to solve the problems, telling them how to annotate the data, how to analyze it. There was someone else having exactly the same problem, and here again myself just answering, so it's something that really works. And on top of that, these guys, they have just published a paper using this algorithm that we were solving their issues a year ago. So it really works, and I really want to convince you that you should shout for help online, because people, it's really responsive. Okay, so that is GitHub. Then of course we have the codes, and you can search on those and so on, but we won't go into that too much. And then there are the wikis where you have all the documentation working. So now, is video working or not? Yeah? Yeah. Okay, good. So there might be. No, just yeah. It's video. But then it's coming. Yeah. Okay, so if I open the PDF file, that now I don't know where I have it. Here. I'm a bit confused with the reward. So these are the exercises for today. So we will go until lunch with this first part, which is more basic image analysis and more workflows, and then in the afternoon with deep learning. So for those who want to run the exercises locally afterwards, or they need to start teaching or whatever, you can just follow these steps. Okay, everything is here, and it's kind of straightforward. Did you all download the data and then zip it? No. So please download the data. So and when you are downloading it, open PG. And yeah, so here it says some tips. So these are some commands that you can touch on the brightness and so on. But there's something that I think is super useful. At least myself, I use it every day. If you click on Control L or Control Command L, okay, you will have your cursor right here in this white thing over there. Okay, and why is this useful? Well, because PG has more than 500 types of commands, and you know sometimes by heart where they are, but not always. So, for example, if you need to do some temporal coding, you can just write here temporal coding, whatever you want to do, and search and just click it and apply it. Indeed, if you, for example, let's say you want to do some tracking and you don't know where is the plugin, whether there's anything to start that kind of thing, so then you can say track and then it will display you all the chances you have for tracking and so on. So I usually use this rather than going to plug in because there are plenty of things. So those are some sort of tips. And now we can start with the exercise. So there's some timing allocated for each of the exercises, and the idea is that I give you that timing to solve it. If you don't have questions, if everything is clear, then we'll go on. If not, I'll share my screen and do it. But the idea is that you always go doing them, and when you have a problem or something you want to discuss, you raise your hand. Thank you. Okay.
So everything in this project, I was here checking the plugins.
Yeah. No, that one is already installed. You have PG? Yeah, that's
I was trying to check if we have
no, some of them are not, but we work in PG is in imaging. Okay, so 15 minutes for the first interview.
Go back.
Thank you. During this 5 years. Yeah, we have a very small year. It's easy not to be in. Because at the moment it's not hard. Okay, time finished. If everyone managed to get the chemograph and the histogram with the activation in time? Okay.
Yeah.
So, do you all understand what you see here? No. Okay. Yes. Okay. So, we have a neuron, and here is all the biology you are gonna hear from me too. It has some gas inflorescence activation, so you can see some kind of bright spots in time, right? When there are synapses and things. And I mean, this is a very common kind of analysis that it's done in neuroscience. So basically, I know here I have a super beautiful spot. So I can just draw it, then I look for histogram, and I plot it. Yes. It was image. I'm going by heart because I don't have the camera. Yeah. So if you pick up a spot that is kind of blinking in time, like that one there, okay, you can see that there's a blinking there, like that, and so on. Then what you see is that you have this kind of activation peaks in time. And this is very common to analyze in neuroscience, like they get many of these signals for each of the neurons, and then they correlate it with some kind of stimuli, or they use it to know what's the frequency of this stimuli regarding, I mean, if it's a neuron from a patient with Alzheimer or not. And there's a lot of math afterwards with these kind of cycles. But this is something quite easy to do. Then if you plot a line instead, because, I mean, these are very thin structures, so you can just want to know what's going on here. And then we move multi chemograph here. Yes. And then the width of the line is how much space around the line I'm gonna take, how many pixels around the line I'm gonna take. I will say just one. Okay, only I'm going to plot for each time point only the lines that appear, only the pixels values that appear behind my line. And this is when I have this beautiful chemograph. So here, if you don't know by heart how to interpret these things, you can take a look at the dimensions. So here you have 27 times 600. And here you have 600 slices. So then it means that in the y-axis, we have each of the slices, each of the time points. And then in the x-axis, we plot the pixel values at each time point. So then what we see here is that around, if I put my cursor here where I have the finger, and I look where I have it, I think it's position y equal 167. So if I go here to that frame, push. And yeah, it's there. Okay, you can see that this bright spot here is the bright spot that corresponds to the time point 167. So then the interesting thing of writing this schemograph, if you are working with live data, I mean, you can, a lot of people does it for acting dynamics, for example. Like let's say that you have the cell control and you want to see, for example, the speed to elongate the protrusion. Then you could plot a line on top of this protrusion and then see in time how it's growing. So chemographs are very commonly used to show qualitatively some of the features that you have in your live data set. And then here you can see all the spots. So it's very easy to see when this specific part of the axon was kind of activated. And then, and this was a very funny thing with Ivan. Ivan said, can we identify all the spots in the neuron, right? And can we segment them? Because at the end, what you want is to detect all of them. How would you do it? So you want to get all these writing things in the image and forget about the rest of the structure of the neuron.
Well, either we do it one by one or we could tell the machine.
Okay, so yeah, one by one, I don't do those things. So how are you going to do it from now on if you need to filter out some information in the image? What do you want to preserve from this video?
What changes through time?
So the brightest spots.
Yes.
So then what do you want to remove?
The noise. You want to remove the noise?
But if I remove the noise, I still have the whole image.
The ones that do not fluctuate. The static neuron.
The static neuron. No? You want to take it out.
Yeah.
How would you do it?
With the subtracting. You lower certain level. Yeah.
Yeah,
Doesn't change.
Right spots. So you need to remove. So remove means subtract. In time. Take it out, right? So, but then you say, okay, I have a video. So something that is very commonly done and it's very useful. You can project the image. So if I say project, this is very commonly also done when you have static things to get the whole structure very, very clearly. So I will just some of the slices and project it.
What is some of slices?
So for each pixel, I have 600 values at one time. No? So I'm gonna sum all of them. Okay. And then I have this beautiful projection of my neuron. So this is the structure of my neuron. So now I can say, okay, then I will just remove it, no? Because that's what I want. I have image calculator. And I say, okay, calcium is subtract my image. And here I won't use float because I don't want negative value. I just want to remove the sign, okay? I say, yeah, I want to subtract for all the frames. I will just change the lookup table so you see something. Yeah. Sorry, yeah. This, I said it was 32, it has to be 60, so it has to be the same field as this one. Sorry, my bad. Okay, and now I don't know how my history. So here, process image calculator. I extract them. You see the spots?
Yeah.
Yeah? But then, I want to see when these spots appear over time, no? Yeah. So then, we can, this is not written in the exercise, it's just, so then we can say, okay, I want to assign a value at each time point. So then I get to know which one the spots are here. So, because I don't know where this command is, I write here temporal. Okay, and then yeah, I can do it. Yeah, nice. So I want this node. You can change the colors you want. You just say yeah. I don't know where here it is. So these are the spots across time.
Like the population of all spots.
Yeah. So I give a value. So this spot here appear at the middle of the frame. These ones at the end, and so on. Some of them might be repeated. So another way to do it is you sum across time, so you want to know how many times they appear. And then of course we want to see it in the video or in the projected cell. So we can say, okay, so now here I want to add an image. So I will overlay. I want to add this color to image. Right, I will put some transparency. 0 is transparent, and then you have it. Okay.
So this is the information time. That
Yes.
one is brighter than time here.
Yeah. So what do you think you have suggested here today? So when we were preparing the exercises, is on the neuron. We can detect the spots. Can we do something? This thing you wanted to do on Monday. He said, can we train a network to get all this? And then I go, yeah, you can annotate for 3 months. And train a network. Okay, so this is I think the reason why you are not doing deep learning now in the first part of the course. So, I mean, this works very well when you have static data. You can remove background, you can remove structures, you can remove artifacts. And that's why I told at the beginning, really have in the hand that you can do these operations, because it will save you a lot of time for quantification. Okay? You don't need to train networks for everything. Okay. So now we go to the second exercise that I think we are.
Yeah. You are talking about qualification now.
Yeah.
But you didn't actually count the number of spots. There are not spots.
Yeah. So you can do analyze. You can now where are my spots? So let's say
do any like orange spots. And I know, and I don't want to know exactly how many you're.
so here you can threshold your image, right? Because you know that 0 is 0, and whatever is the full 1, so then you can threshold. So I need to convert it to phrase here. Okay. With threshold. And we say whatever is below 1, we get it. Okay, right?
That's all.
And then you can do something that is called analyze. particle, and then you get the particle.
I usually do that for 5.
But then you can choose,
As the option.
so you count, and then you can get, for example, I want to analyze this one, and then you project this Roy in the other one. Okay, so you can get this Roy. Yeah, made up. I don't know which this one, but I can here, okay, so I can copy the reading. Let me see if I manage to get one. And one. This one, I just copy. I come here. I paste. And now we do the when we did before, that was plot profile, and you have the assumptions. So we can go for each of the data outputs. So you can jump on your image. Okay. So how for this one you only need 5 minutes. Okay? So what is first? You need to put something like if you do something bigger, it's not there's something that is all these combinations. It's very big. It's important sometimes you have the next so and then you might and then you have many different combinations that it's not that straightforward. So here is when this is and you can find the things that you are very no, in this case, no. I mean, unless, for example, if we have a huge but extremely huge image, we won't have it like we have evidence 20%. So it's going to dimension. Now, well, then maybe you need to choose a very easy one, but seriously, you try to like choose one and then so raise your hands when you are finished, because yeah, okay, finish, yeah. Okay, so maybe we can move to the next one. This was easy, right?
Yes.
Okay, so just move to the next exercise because I think it's starting with a little bit more interesting.
Yes.
32 bits.
Yes.
Okay, I will explain it. Okay. So here there's a very important question, and is what is 32 bits? So bits is what we use to store values in a computer, right? We usually use the binary codification like 0, 1, and then, for example, the number 2 could need 4 kind of digits and so on. When we say 32 bits, is that we give enough memory to the computer to compute float numbers. So now we are not going to use 0, 1, 2, 3 until 255, but now we are going to start using double numbers like 0, 0.3, 0.13, and so on. Okay, and then the issue is that when you use 32, then you can have any value, and that is what we usually use to do math with images, because we want to compute numbers, right, and normalize the images and so on. But when you want to quantify your signals and work with the data that goes directly from the microscope, you need to see if it was 8 bits or 16, so you preserve that scale, right? In 8 bits, you will only have values from 0 to 255, and in 16, from 0 to 65,000 and something. And if you are going to compare two images, you need to make sure that they are in the same value. So my recommendation is usually to normalize always between 0 and 1 and use 32. But you do it always for all the images, all the bunch of experiments you have, and always using the same normalization. You cannot do ones with percentile or whatever. I remove the first values close to 0, and then in the other one I do a standard normalization or whatever. You always need to do the same one. And then when you have the fluorescence, everything goes from 0 to 1 in your experiments, and you have it controlled. Okay. Yeah, because it should require. So, like, we get Now I look at okay. But then you should have. I should, yeah, yeah. We were trying to circle I'll show you now. Okay, so did all of you manage to not the exercise finished? Okay, so I saw some of you that were struggling with their responding. Yes. Right? What happened? Why you didn't like the responding? I don't know. What happened? I just don't finish. Right. Okay. So, and why is this? Why do I need to adjust the contrast? But why? What are my pixel values here? 0, 1, and 2. But how many possible chances do I have in the image? 8 bits, so 250. 56. I have 256 possible values to display, and 0, 1, and 2 are very close to 0, right? They are very dark in my display. So then I need to adjust it, right? And just say, okay, from black to white, you only concentrate in these pixel values because I don't care about the rest. And that is what happens when you transcode the image, because you are putting, just saying, like, I don't say apply, okay, I don't change the pixel values, I just want to change the display, but not the pixels. So then when I go into threshold, some of you were getting this, right? But if you click on, 0, yeah, okay, so here I'm only taking the background, here I'm taking the boundaries, and here I'm taking the entire image, and this is not what I want. I want to, for example, this. It's not letting me, so I will just do it here. 1, 2, and 2. Okay? And that is the bit that you need to play with the numbers and the display. Did you all manage to get the colorful image with the watershed?
No, the problem.
When I apply but did you run the watershed?
Yes.
And you got like a grayscale kind of image?
Yes.
Okay. So, this.
Yeah.
And you want colors? Yeah. And what were the colors?
All of them. Attribution.
But what's the name of the display? Look. Up. And then what we can do to find the look of it? Functional. This table, so it is LUT, but I can start playing it here. And here we see where we have it, okay? I can just run this one if I want. And we can just say, lookup table is LUT. So I can just go to the wikis. Display lookup table. So these are all the chances we have. So here you see lookup tables as well. Image lookup table. And then you choose whatever you want. For randomness, you can choose the glass wave, for example. This is and so on and so on. Okay, and we can be playing with this for the entire day, but we don't have the entire day. Yeah. In this one, it's the one, but then I mean the other ones, if we choose eyes, for example, then it will it will go like that, and if you want to see actually the color bar,
It's always random. Right?
No, this one is not random.
So
This one is organized.
but I mean, why are you choosing the why did you think it's organized? You're not choosing get number of bacteria to be like
So what did you get in the image? What is this image? What are the pixel values?
0, 1, 1.
No. Why did you do the watershed? Okay, so from now on, when you do image analysis, you don't have bacteria here. You have pixels and values. Okay? From now on for your entire life. And so now, what do we have in this image? Pixels, very well. And numbers. What are the numbers in this image? That if there were 0 and 1, then I would be seeing just black and white image, or 2 colors. I've seen many of them. Why do we have different colors?
To be at your price.
Because we have different values in the image, right? If I put my cursor here, you can see how the value up there is changing, no? So what are the values telling us here? Each different cell has a different value. Why? And that's why we did the watershed, because we wanted to identify each independent cell so we could extract measures for each independent cell. So then it's not random values. Each cell has one value, one unique label. Okay? And this is called instance segmentation. So this is not binary. We went from a binary image into an image where each individual has a unique label, so we can track them, we can analyze the morphology, or we can just count how many objects we have in the image. So then when we choose the lookup tables, it's not random. Here I'm showing you this one, the eyes. So 0 is green, and then this maximum is red. So this is the maximum number of pixels values that I have. If I want to count how many bacteria I have in my image, that is what you did with morphology. So you calculated the the analyzed regions, right? Let's compute all the metrics. So here it will tell me, like, these are all the different regions that I have labeled. So you can export these into an Excel and get a number of elements you have and the pixel count. So for example, I know, let's say I want to see this bacteria has value 10,251, that's the pixel value. So I go here, is this one, okay? And this is telling me how many pixels it occupied. Of course, it's the same as the area. Then we will need to multiply by the pixel size, but I'm not giving it to you in this image. The perimeter in pixels, the circularity, the bounding box like the X and the Y, and many other metrics that we have here, okay? That might be of interest for students. But this is not a binary image, okay? Yeah. And then actually, if you want another way to get this, is by analyze particles. Yeah. We need to go with this one. I will have here all my different activity, okay, that I can just choose and get some metrics, okay? And we have a total of 100. Okay, so yeah, no. So now there's a homework there that is about how to do, I think, some automation of the code that is with image and macros. And some of you used macros, but most of you didn't, right? Okay, so here, all the steps you follow, we can record them in a macro. This is an amazing thing that CG has. And we can go to macros, record, and it will start translating into code whatever we're doing. So then you just need to copy paste and run it on new images. And that's how you automatize the analysis. So we can, if I open this image, then here you see what's the code to open an image. If you duplicate it, you see the code for duplication. Then I choose this image, and if I press code, so one was 1 and 2, right? And the other one, was I was one. Yeah. And then I do the watershed from Morcolic chain for this one. And I say I want to segment this one with the markers from testing. This is all what you did, the mask. And I hope this one works. Here it is. And now if I go here, this is the code I can click on create. And then we have some code. And if I go to this now, I just close all my windows. Okay, the code has also the open one. And if I run, probably it contains at some point, but it's something you can see. It complains, so probably it didn't duplicate the image. So, this focus search bar shouldn't be here. Yeah, so it's really recording whenever I go to this white thing, so it shouldn't go there. And now it should work, I think. Authenticating. Yes, but this close the exercise. So you clean. I mean, we have the bottom in the solutions, okay? But basically, it will complain that you can run one by one selected code until you see that everything is working, and then the same steps you follow. But now it should work, I think. Yeah, there it is. So now in the homework that we want to do now, it says that you have a folder with many of these images that is called boundaries, I think. So what would you need to do if you want to automatically analyze each of the images in the folder? What do you need to add to this code? a directory where you store the images. And then
When to open, like,
that is here already.
Maybe
The loop, yeah, the loop. So you need to add a function that will tell you how many images we have and their names, and then you write the loop. For each element in this folder, please open, apply, save, close. And then it will, and you have the solution there, it's written, so you can use it whenever. But then also, here when you are typing in the code, it suggests you many things. Okay, so it will give you I don't know, the whatever you need. If you say threshold, it will suggest you some functions, and there's a lot of documentation everywhere. So I don't say that you're gonna be expert programmers in 1 week, but I think it's something very doable to do regularly. Okay. Okay, so now I did that code, and if you do it properly, you should get a folder like this one with the instances. Okay, so where each, so if I drag and drop the folder, you can see here the video of the bacteria growing, and each of the bacteria has a unique label. But what would you ideally do with this? What can you do with this kind of movies? This is the second, the next step. We can do that. Let's do that. Yeah. Leave. The stack. No, but so now we have the stack analyzed, and we have some video time lapse where we see bacteria proliferating. So ideally, what should we do with this?
You can follow and track.
You can track the bacteria, exactly. So let's track it. So then we go to TrackMate, so we look for TrackMate. I don't like these colors because by those elements I'm not changing them. TrackMate. And then it will ask you to change the TS time, so yeah, and what to track. And then here you have many chances. So we use the label image detector because we already have each of our cells with a unique label and properly segmented. But TrackMate has other chances, like use some internet models or some thresholding if your image is raw, like for example, if you have particles to track, it works quite well. So I will do this, and then I do next, and then you say preview, and it's showing the first time, the first one, sorry, preview. And here you see that it properly identifies each of the bacteria. So I go next. Now it's identifying each time point. Next. Yeah, our labels, our data is process, so we don't care too much about it. And then with the lab trace. A lab trace, simple lab trace. And this simple lab tracer doesn't consider cells splitting because particles, they don't split. Okay, so if you want to track particles, if there's a new spot, then it's a new track. This is one chance, but we don't want that one because our bacteria, they actually split, right? So then we can choose the lab tracer. And here we can say, okay, the links can be, bacteria are quite elongated, so we can have tracks with the distance from each time point of 30 pixels, for example. Here is, do we allow closing gaps? And this means that I have a bacteria that appears here, it doesn't appear during the next frame, and then it appears again. Should it be in the next track or not? Because our images are very well segmented, we say no. But sometimes if you have blinking things that they may oscillate in time a little bit, maybe you want it. But this is really up to you. I don't know. It depends on the use case. So in this case, we don't want it because bacteria are very well segmented. And of course, we want to allow the splitting. And the split, well, it can happen again with a distance of 30 pixels, for example. So this is a bacteria split into two. And we click next, and here it will draw the tracks of each of the objects. And it considers that if it is a split, it belongs to the same initial track. So it's like a branch. So here we see some tracks. Some of them are correct, some of them are wrong. We can actually display with the speed. Like to check which are the fastest ones or not. And then here as well, and then we can see the branching. Okay. So this is how TrackMate works. But then I have a question here for you. So we go here, for example. And there's a split here, okay, of these two bacteria, but that's like in your track. Maybe I can display this in a different way. Yes, like this. So then it will draw as long as you close. So here that we have different splitting. For example, here is deciding that this one is splitting from this one. There are many errors, right? So then, what would you do here to start? Get back to try to see if it's. But there's another issue here. Can you follow yourself what's going on with all the bacteria here in this kind of for example, so here I have these two big ones on top splitting, and then this one is splitting as well. And then is this one coming from here, or is this one coming from the one in the upper side?
Can you show us the one before? Again?
Yeah, or no? What about, I mean, it could be mathematically. That's not correct, because it could be that this one moved up and got short and it got shorter. So you know that, but the program doesn't know. And there's a way to overcome this limitation. Indeed, here is even more complicated. I will go from this very view to here, and solving this linking is not as straightforward. There are different possible solutions in time. Why do you think it is?
Maybe you should have required the infinite with shorter
Exactly.
more frequently.
Okay, so when you're working bacteria, it's super tricky to track. It's one of the most difficult problems, I would say. And it's because all the shapes are the same, they like to be in colonies, and it's very difficult to assign, right, if a new cell belongs to the one in the right or the one in the left. So when you want to do live imaging, you need to adjust also very well the timing, because if you want to track, you really want to see some overlap, be able to follow without ambiguity what's the proper track. But then with bacteria, there's something that is very cool. You usually don't want to track. What do you want to do with bacteria? Better. So you want to analyze colonies, right? And the growth rates. No? Not that much the tracking. So then you wouldn't need tracking. No? What would you?
Count.
Count on time. Exactly. So you count the number of cells you have in time, or maybe per area, right? You could say, okay, here I have very clearly one colony here, and then another one, so I can identify colonies that are very big at the end, and track how this area changed over time, rather than for each cell. And that is part of, that's when I said at the beginning, you need to be creative. You really need to identify very well what's the objective of your quantification, and not do segmentation and then tracking, and then I spent all my life trying to get a proper track. Okay, good. So then this one is solved. Now we go to the funniest part. That is registration, right? No? Yeah? No? Yeah. What is registration? Why do you think I'm showing you this thing? No, I didn't say it in the lecture. Expansion microscopy, because you read it. Okay, so just before that, do you want to do a break? No? Go 10 minutes, and then we keep until lunch. No, we have time. I don't know how time it is. No, we don't. This is great. Let's run it. Okay, so yes. You want to turn off? Please? No. But there was too much living in the class.
Okay, switch that up, there's no computer.
Yeah. If you want to open the windows as well. Yeah. Okay, so segmentation was about quantification, okay? But we can do many other things, and I don't know how to put this without giving you the answer. But so, who is doing live imaging here? Or 3D stacks? Yes. How does your microscope do live imaging? I mean, this is my, you put the finger, guys, the ones who, so now this is the objective. How do you do it? How does live imaging works?
Objective.
Yeah, this is the objective. So I have my wheel, right? And how do I usually do the experiments? Like this, no?
If you're doing multiple positions.
Who does live imaging? Pedro, you don't count. Anyone here doing live imaging or planning to do live imaging?
More than 30 minus.
You all fix your samples.
Ever.
And the objective.
Oh, like this. Okay, but you don't spend the whole day doing one experiment just to get one field of view, no? Okay, so then how does it move like this, no? And then gets back. Okay, I'm back. And does it always get back to the same position? Yes. Does it?
You check.
Yeah. To the exact same position? At the nanoscale? So then what happens usually with your,
Because that's really, there is some drift.
there is some drift. And that drift, what is the effect of that drift?
Distortion.
Distortions in the fluorescence, in the time lapse, in the tracking, and many, many things. So then what do you do to correct it? Align. And that is called registering images. Okay, so that is very common, and this is what we usually do here. So usually in 3D, especially if you're studying, for example, organoids, and you're doing live imaging with multiple channels in 3D and time lapse, what happens is that you need to register, because although the microscope is calibrated, it doesn't really get back to the same position. I mean, it's almost impossible, right? It's a mechanical thing. So usually we have some kind of vibration. Okay, distortion in the image. But more interesting than that, when do you think you can also need registration? So now register means to identify one image into the other one. right? And get back to the same point. Whereas in fixed samples, you fill I'm very right imaging, okay? So when you fill the samples and you fix them, and you do other type of microscopy that is not light itself, electron microscopy. Does anyone here do electron microscopy? No? But do you know how it works? So you slice, right? And then you fry with the electrons, you get a beautiful image, then you slice again, and then you want a volume, but yeah, the volume is in your folders. What do you need to get the volume? You need the step, you need to register again. Because of course depends on how expensive is your microscope and all the equipment it has. You usually do it manually, like take an image, then you go again, and that's how you get all these neural reconstructions in electron microscopy. Then what else? What else do you think you can need registration? Histopathology? You slice by hand, you break some hands in between, and then you try to get the whole volume of the tumor, for example, right? So there you also again need to register each of the slides against the cementation. So what else do you need to do registration? Then, do you know what is PLEM? Correlative light microscopy. But you mix fluorescence and electron microscopy. You didn't see anything like that? I'm sorry. But tomorrow, you say, what do you use to register your images? Because I'm going to show them super well co-aligned. And that doesn't work, because what you do is you go to the fluorescence microscope, you get your white field image of your proteins, whatever channels, and you go with the sample to the electron microscope and you fry it completely. And then, of course, the points that you got with the fluorescence and the one in the electron, they are not the same, and you need to co-align them, right? And then it's even trickier, because the electron microscopy has nanoscale. The white field doesn't have nanoscale, it's microscale. So how do you know which of the exact proteins you're imaging is the one that was emitting fluorescence? Okay? So all these things, and then of course, because you're trying to make up this kind of deformation in your images and so on and so on. So registration is usually not very much taught. It's a super complex kind of image analysis stuff. Math. But there are tools to work with it, and I don't want you to know the math, but I want you to know what are the kind of registrations you may need and what you can use in future. Okay, so there are two ways. There are linear and non-linear registrations. There are rigid, non-rigid. So linear, it means when we have our image and we don't really apply any kind of elastic thing, right? So we can make it bigger or smaller. That is what happens. For example, no, I'm not going to say that. So it can be bigger or smaller. It can be that fine. So the parallelism is preserved, but it's still a little bit shrink. It can be translated like the drift. So this is rigid body. I have exactly the same thing. It's just that the microscope was moving like this, and we need to do that. And then the very more complicated ones are the non-linear. When you destroy a little bit the uniformity in your sample, this is what happens with histopathology, with PLEM, with electron microscopy, and with expansion microscopy. Mario yesterday gave you a talk, and he mentioned something about this, right? So expansion, is it uniform or not? Should. It should. And how do we measure it? No. So what did you tell me yesterday?
We need to acquire before expansion.
Yeah. And then? Just look at it. So until now, people were just looking at it, and today I'm going to show you a little bit of how you can show in your papers, if you use it, how to measure or quantify if it is isotopic or not. Okay. So the first exercise is about correcting the drift in one of the videos that you opened before. So it says 10, I will give you 5 minutes, and then the next one, I do it with you, okay? So try to do the very first one, and you see what the makes it. Okay. Yeah. You already did.
Yeah. Yeah.
Yes. And then of course the pictures in that's when it's hard. Okay. Yeah. 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 One of the images that you should open is this is the live on programs. I think I didn't export it correctly. So here is the drift. This is at the very, very high resolution. So the drift is much higher because the error you can see. So this basically they use it in spectroscopy. So then when you correct it, this neuron is not really moving. Okay, this is a neuron, they don't move. Okay, it's static. And what is moving is the object. Okay. So
Almost correct. Yeah, so yes, neuron stuff.
yes. Here you see, okay? So that's in reality the neuron, and then you see the activity, and then it's not moving, so that you can already compute, I don't know, whatever measures are on this. Okay, so I guess we all managed to correct it. Yeah? Okay, any question? No? So this is very cool, but usually what we want to do is registration in PD and across channels, because of course to compute the registration we always compare against something, and if we have, for example, only spots that appear very sparsely in our movies, the registration won't work because there's nothing to compare with. So many times in this situation, what researchers they do is they acquire different channels, and they acquire a channel that will level some structure that doesn't move that much in time, so we can always compare against that and register in time. Do you get this part? Let me open. So if we open the next the data for the next exercise. Let me yeah. So these two beautiful images. So this is some mice lung vessel, okay? And the vessels, they don't move much across time. So this is the 3D time-lapse image. But of course, what we want to see is cells, right, in these vessels. And this is what you see in the other image. So this cell, it's moving. And if we try to register this, is the 3D, you can see there's some drift here, no? Can you see? Maybe not, but you will see that. So here, the thing is that we assume that we are going to have drift, because the microscope, what it's doing is it images. Okay, so this is the volume. Okay, so it takes one image with with channel 1 and one image with channel 2. It goes to the next slide. Channel 1, channel 2, channel 1, channel 2, channel 1, channel 2, the whole stuff. Then next time point, all the channels for the volume. So then there's a drift between each time points, not only for one stack, but for the whole 3D volume. And then we know that our cell is going to be moving, so if I try to register it, it won't work because it changed its shape. So then they acquire also the vessel, and we can register everything against the vessel. That will be a static kind of a structure upon time, and then we can apply this same correction that we did to the rest of the channels, and that is called multi-channel registration. Yeah? So that is what we are going to do. So now you see whether there's a drift or not. There are many different ways you can project it in time. You can, there are many different ways to do it. Here I suggest you some just to play with colors. That is, here you can display the cell with the orthogonal views. It's written in the PDF I were to go for the orthogonal views, but it's not so right. So if I'm here and then I move across time, you can see the drift, right? It's very clear. There, yeah, in this orthogonal view. So this we are seeing X, Y, but also C in this part. Okay. But also we can color code it, and we can say, okay, I want to just say right here. Temporal color code again, in this one. Okay, and you can see here, right, everything should be in the same color and in the same position, and there's some drift that appears from 0 until the end of for each cell slash. So I want to correct this, and there's a wonderful plugin called Postdoctoral that we can run it. So I think we don't need to open the images actually. So we say plugins, Postdoctoral time apply. So we want to, time estimate and apply. And here we so we can just take one and drag and drop it here. Okay, so I will estimate the width in the vessel that is called Mausland Epithelium. And I will correct it, and I want to do it in the X, Y, and Z dimensions, okay? Because we have the 3D as well. So we just click on OK, and it's doing it. Are you running it?
One file per time for the plugin to run.
Is the vessel's file?
Yes.
Yes. So when you finish running, it will create a new folder where you have your file. And here it has the grid plots, like how was the grid across the time in X. How was the grid in C, that it was increasingly. And this, can you think why in C is like this? What do you think it's easier to get back to the original location like this? Or not only get back to the original location, but even in C, be able to manage it. That is even more complicated. So that's why you see C liquidity is also huge. Okay, so once you have these files, the very great thing about this plugin that is not that straightforward with the rest is that you can say, okay, now I will give you a new image and I want you to apply these changes that you calculated in this new image. And that is the plugins, passport degrade, time apply. So we don't need to estimate anymore. Yeah, so now I give the file of the cell. And then here I choose the grid correction file, that is this ESB file that is telling what are the changes we need to apply. So when you have your image, you create it in a new folder with the name of the image, and then you have all the parameters and the results and the corrected image. So then here we say, okay, the where to save. So I want to just save it here. Thank you. Okay. And I click on OK. And it will correct the cell image. You don't, you need to wait until it says like, and I go here, and there is the corrected image, XY and XYZ. Do I manage to have your images corrected? Oh, wow. Still thinking? I would like to write. Again. Sorry, I don't know. So sorry. Which is the same that you apply, don't you calculate? You apply the transformation that you just calculated. The other one? Yes. No, I'm not sure. Yeah, it should be, but I'm not sure. So who didn't finish yet?
Yeah.
Aha. So in the folder you will have the images corrected. So you can do the same as we did before to just if there is drift or not, like color coding and projecting and those sort of things. So here are the files corrected. I also gave them to you so you could play with them. I converted them to 8 bits because otherwise they were super heavy. So some of you were not seeing the images because you needed to adjust the brightness again. But well, here are the images already. So if you match them, okay, we can say like match channels. So I want to put in green cells, for example, and in gray I want to preserve the vessel. Here's a way to do it by matching the channel. So now here I have my beautiful cell entering the vessel and embedding the lamp. And you can do some 3D projection that I want to do because it takes a lot of time, so I will just show it to you. I have it already computed and you also have it computed in your data. So here is the cell, the drift corrected, and we can actually see in time how it's entering the vessel. Okay? And if we go deal with the uncorrected files, we can see this quite clearly, so you will see the things are vibration. So any question here? No? Okay. So then I think we will move to the expansion one. That it's a bit more complicated. Right? So image stacks like 3D, but otherwise just like 3D. But it takes like 10 minutes. Oh, good.
Yeah. I don't know about MCG.
Yeah.
We thought that it was computing, but now I don't
Yeah, I think you're okay, so the expansion microscopy. What I'm going to show you today, there's one part that is quantitative, there's another part that is not that much, but there are no straightforward forward ways to compute this. So we kind of, with Milo, came up with this workflow that we thought it could be useful for you. But Pedro is also an expert in expansion microscopy, so if you don't agree, you can complain or provide some other helpful approach to follow. So the idea, if you program in Python and in MATLAB, there are libraries to put this a little bit more automatic, but then it also implies that you need to know programming. So that's why it's going to be a semi-automatic kind of thing. Okay. So the idea is that we have this pre and post expansion data. So this is the post expansion and this is the pre. And what we want to measure is how much the structure was deformed after the expansion process. Okay, just to make sure. So this is more like a quality check rather than a quantification of your results. So we want to check the isotropy of the or uniformness of the expansion. And this is 2D, but of course you can go into 3D. So first we need to locate where this structure is in this below. And that is the non-automatic part, okay? So we know that this is here, okay? And there's again one plugin in FG that is called Rec. That it can provide you different kind of transformations, and from all these 5 here, what do you think we need to look for this example? What is the kind of transformation you need to do in one of the images for localize? Okay. Scale, because it's zooming and out, and what else? Rotation. And rotation, right? Because it's the other way around. So we go to scale rotation, and then here you will see 2 dots on each of the images that are super small. So one is green and one is yellow. So this is the green, I'm moving it here. So I just put the green here and I place the green in my other image in the same place. Okay, I need to tell, okay, this structure here is here, more or less, and then the yellow one in another place. I will just place it there. And then the yellow one here is here. So then I'm giving the algorithm these 2 points that you see here are exactly these 2 points that you see there. And it will just need to calculate what is the zooming out index and what is the rotation. Once you have it, you click on accurate and automatic. So this is a non-automatic one. Okay? And it's a bit there's no code. In MATLAB specifically, there's a very nice code for this, but yeah. Did you manage to have this one? Yes. Do you see that point? Oh, yeah. Okay, so now this is what you need. So I agree with the yellow
Yes.
And then the yeah, so this is a bit tricky. There are other ways, but not always what was, so I prefer not to show them. Okay, so when you have these two, what do you think is a nice way to see if the structure was deformed or not? What would you do? But what is the picture? What is the size difference? No, the size because you said like okay, so if I want to see whether my structure was deformed or not, now that I have both of them in the same place and they are supposed to be the same, how do I compare them? Overlap? What else? I can just subtract them, no? If they're the same, it should be 0. Yeah. Right? Okay. So we can, if I want to do subtraction, because very clearly they are not the same. So I will just merge. Oh, you need to duplicate this one. This is a stack. So you need to take the first one. So we merge, and the red one is the post expansion. If there is an electronic gray, for example, is the pre expansion. I will keep both. I overlap. The answers are not the same. Okay, so here is the overlap. And very clearly they are not the same. Okay? Do you manage to overlap the images? I think you need to convert both to the same slide. As this is quantitative, you can change. Okay, you can put them in 8 bits, each of them. Okay, Okay, so there was a question here that was asking whether we should get the same images or not. This is the pre is in red, the post expansion is in red, and the pre expansion is in white. Do you think they should be the same? In theory? Yeah, right? Why they are not?
Maybe it wasn't quite homogeneous in the experiment.
Exactly. So the expansion is not like working precisely super well. But then also there's something else that in this case I didn't have the reconstruction parameter. So this is the stored image, and I know that there's some astigmatism, right, when I apply the images. So it might be sometimes that in the edges keep changing a bit, right, while in the acquisition, because of the like super resolution that then we need to reconstruct. So usually we try to focus especially here. And it feels that the image was kind of a stretch. So during the expansion, it did it in this way, but not, if it was uniform, it just zoom in and out. And it's more like stretching like this, so it's not the same. Now, let's see how we can show this in your papers, right? So, a very common kind of application for registration is not to have things in the same way, is to study what was the transformation among them, okay? So now what we are going to see is how we should transform the previous image, the pre-expanded image, to get into the post. If we know how we should transform it, then we know if this transformation is isotropic or not, okay? And that is usually, it's very commonly used to do quality checks. Okay? It's not for quantification of your results, but just to know if things work or not, and until what extent they didn't work perfectly, because things don't work perfectly, I think, never. But maybe it's admissible, the kind of change. Okay, and that is what we really want to see. So now there's another planning. So you have both images with 8 bits. This will allow us to have more memory in our disks and make things faster. So we first need to normalize the images. So we are going to put the intensity values in the same way. So there's like a macro file that I gave to you. Is this one. So we will just choose one image, run, choose the second image and run again. And what we did was to convert all the pictures to the 0-1 values and then multiply it by 255, so it will convert to 8 bits. And this means that the brightest spots here are also the brightest spots in the other image. So we kind of put both of the images in the same range of values. This is a normal practice. If you want to do some math, you should always normalize your images before, and then you make sure that things work. But this is only for the math. Then for the multiplication of fluorescence, the original fluorescence assignment. So then again we go to registration and we run B and work. And then here, please put exactly the same parameters that I put you in the PDF file. Okay, because it's a bit complicated, the registration. So when you do it in your own data, you may need to run it several times until you see that things make sense. Okay, so here the source is just check it. Is the post expansion, so is the reference, and the target is the data. In my case, if you open the file that I give you that is registered, it's the pre-expansion. We want it accurate, and we are going to downsample a little bit because we don't need all the resolution for the registration of the structures. Then here we go from very coarse to super fine. This means that we are going to run an optimization algorithm, and the way it works is that it takes one image with its normal resolution, all the pixels and very big, and it compares the difference. And then it says, okay, I need to adjust it a little bit, and it adjusts it. And then it goes to slightly fine details, and then try to adjust this one with this one there that it doesn't exist, and it will go finer and finer and finer and finer. And it's an optimization. So there's no correct answer. It will reduce the difference between both images until a certain threshold that we put it here in the what did I do with I think I lost all my I think I hit. Okay, I will close everything that you can put the parameters there. So these parameters here, the first two is to preserve the shape of the image when we transform as long as we pick a video we see what meaning, and the rest we let the same, we say therefore, and if I click OK, this is what happens. So the process starts trying to co-align one image and the other one. And when there's no much change and much error, it will stop the optimization. And here you see the kind of deformation we are doing. So this kind of processing, they are not exact operations. So most of us, we won't get exactly the same grid, but it should look very, very similar. We can also stop whenever we think that it's done and it's good enough that we are happy, we can click stop also. Yeah. So here I have my register images. So here it modified the pre-expansion, so they look the same. And here I have the modification that I have to do in the image in order for both to look the same. Okay, so here you see in the center, they are modified. So here there's some kind of pattern. Do you think the deformation was isotropic? No, not at all. So there's a paper that was published 2 days ago. They are doing exactly this, and they have some plugins in Entiometrics if you want to go through that. It's the same idea as this. So they basically measure how is, like with some numbers, like how uniform are these deformations, and you get a quality check for your expansion. Okay? But this is a very nice way. Actually, these ones here, we can avoid them, but this clearly is showing how our expansion did this kind of process, right? And deformation. So with this, I don't know if you have questions. Of course, this now the question is it important for our sample? Do we care about it?
Yes.
Yes. Why? It depends on what we want. It depends on what we want to measure. So things are not black and white. Things are not good or wrong, and less in biology. So we have microtubules. What do you think we want to measure with that image? Length, size. Will this deformation change a lot? Because now it's not isotropic, it's super long, but does it matter? For size, do you think for size it matters? The length will change that much? The distribution of the microtubules? No, right? They have the same cross-linking, the same sort of thing. So, does it really matter?
Maybe not in this case.
Not in this case. So, that's very well. So, for example, when they are expanding brains, that I don't have the data, that is 3D. So when we compute this, imagine that locally we are actually changing some things, but overall we are not changing much, so then it doesn't really matter. Now, if we want to use this expansion to see how was the connection between neurons and the synapses, and we are breaking those, then it matters. Then we cannot use it. Okay? So that is why it's important some sort of doing this and checking how things change, but also not forgetting what is the objective. Okay? So now finally, we want to see how much resolution we want with one image and the other one. So one very nice thing we can do, where is my image? So I will just crop the middle part because I don't care much about the boundaries of the image. So here, right? And there's a plugin and there's a method called Squirrel. That's maybe Pedro can say more than I about it.
Well, yesterday, Hannah briefly mentioned it.
Okay, so Squirrel is a method that will tell you with a reference image how much resolution you want. You cannot do that without registering the images, but now they are registered. So that's super cool because we can see how much resolution we want with the expansion in numbers. So if we crop the patch, and I will just duplicate the first and the second one. So yeah. Like here, and here. And I will close the rest of the things. I have these two, and I can say plugin nano.j. And we need to run the unicorn one, that is the last version of it. And then the reference image is the registered target, and the super resolution one is this one. You can see the names here. So we say, okay. And then here we have some sort of error map, okay, with the pre-expanded and post-expanded. I mean, actually, there is very nice spots like here, okay, that they are different. Also here where you can resolve those microtubules super well here, and here you couldn't. So, I mean, there are some sort of things that you definitely gain with the expansion. And then it also gives you some numbers. Okay? Like the Houston correlation between both images, they are very highly correlated, the scale error, and so on and so on. Okay, so this is more quantitative, and it's what you do when you manage to register things. Okay? So now, the last part is what can you do with this expansion microscopy, and this is because I love it, that's why I put it in the exercise, but it's a very, well, you can of course go put it with colors and then see very clearly where they agree or not. I think it's a nice one. I don't know. And then what we can actually measure is the orientation of this microtube. And now with a very nice grid. So we can click orientation J, analysis. So here I want to see the color, I want the gradients in degrees. And this I'm going to leave it like that. So when we run it, we get this image. So okay, so imagine that 0 is like that. Okay, so the color is telling you how much you need to move, how many angles to get into this position. So the vertical ones are in red, then these green ones are like this, okay, and so on and so on, and then you can get a distribution of your angles in the image. So we can threshold this a bit, so we get only the area of the microtubules. And then we can see this is the histogram, and we can just ask, okay, what's the mask you are using to compute and over which pixels when you threshold this coherency, and then we get this histogram. So most of our microtubules, they are in vertical position. Yeah, so they did make sure. And then there are some like this one in horizontal, is here, and some like this here. And I mean, this is something super nice to do with microtubules. For example, if you have some mass of cells that they are moving and you want to see if they are oriented with the flow or how they change, if they are aligned or not. Yes. And yeah, if you track the orientation upon time, you can also see alineation to mitosis, for example. And so yeah, I think we can go for that. Okay. So. So, I hope you got a fresh mind. I'm not sure because now we're gonna switch the topic a little bit. So, in the first part of the lecture, the idea was to, and the practical sessions also was to give you a glimpse of what is biomedical and why it is important for biology, or know what are the considerations we need to have, how you can play with it, because at the end it's our playing, right? And I hope that you saw that there are many steps to do different things in terms of quantification. And now this part of the lecture, it's, well, as promised, is about deep learning, so it will teach a little bit without a very dark link, but your interests, right? And the fact that these tools are super useful. I think same as using, I don't know, some statistics, it's good to know about them. And it's also to get an idea of how they work, so you don't make too huge mistakes, right? So while using them, and also to give you kind of independence, analyzing your data, and also strength, right? Because this at the end is for you to do your research with the highest quality possible. It's not just because it's fancy, okay? So, with that being said, I think I already convinced you that there are many different places in the, let's say, in the research work with microscopy images where you can apply biometa-analysis. And as I told you, it starts from the beginning. It starts from the acquisition. And this is also what we call the deep learning playground. And one of the very important things is that to use deep learning, we need data. Okay, that is one of the very strong points here. And data is acquired here. Sometimes even the ground truth data. So if you want to use this, you need to think it even before starting your acquisition, right? Because you really need to think very well how you should acquire the images and what you want to exploit afterwards. So now let's go bigger on this. So in classical image analysis, what we usually do is we define equations, right? To get out our images or to do some transformations, and then we apply those very, very fixed and very well established equations to the images to get some results. In this case, is to super resolve this image and denoise it. And when we use deep learning, this changes a little bit, right? So So, and this is not only deep learning, also machine learning. What we do is we give data to the system. We show some examples of what we have and what we expect, and the system automatically learns what are the transformations that need to be applied to this input in order to get this output. And once we have shown many examples and we go through this training step, we get the predicted method. And here, is this next completely? I'm sorry. So here, why data is important, right? So imagine that we are trying to learn or trying to teach that this image is a tomato, right? And the system will take these kind of features and will learn like, okay, this is roundish, this is red, this has small texture, it's a tomato. But if we give this new image that the system didn't see it before, it will also classify it as a tomato because it's round, it's red, and it has like a small texture, but it's an apple, right? So that's why data is so important. So basically, what we do in the case of medical image analysis is we provide this example. So here, for example, we have biopsies of a patient with some sort of cancer, right? And biopsies of a patient that is healthy. We give them to the system, and then we have a smart system that is able to do the diagnosis. So that's basically the idea, and we now go a little bit into that, and this is really this organized. So the reason why at the beginning I was trying to show you what was a convolution is because the networks that we use in image analysis and all this deep learning stuff, these models, they use many different types of convolutions. So that's why I wanted you to get the idea of what means filtering an image. So now the difference is that we don't define what are the filters, but the networks, they will learn what are the optimal combinations of filters that you need to apply to the image in order to classify it. And this is what we call convolutional neural networks. So very, very briefly, we have an input and we have many layers that compute many of these filters many times and downsample, upsample the input until they get some sort of vector or features or like matrices with values that allow the network to do the classification. So then what we need to learn is actually which are these feature filters, right? This is what the network learns or what is trained, and these are called the weights. So when we train the network, so you can get a bit of an idea of what this means, what at the beginning we train, and these filters, they produce random things. But when the network is trained and knows in this case how to segment this image, if we go to each of the layers of the network, we start getting many different features of the image that we don't see in the input one. So here this is a unit, it's a very well-known architecture for image processing. So in the first layer of the unit, if we plot some of the features, they look like this. So these are different features of this single image. If we go deeper, this is the structure of the unit, so it downsamples and it filters again. It downsamples and filters again. And these are some sort of features that the network will combine one with the other one to at the end produce an output that has the background in kind of whitish, the interior side of the cell in whitish and black, and then the edges of the cell. So these kernels are what we train, and this is what is called weights, each of the values that we have there. So when you hear that somebody is training the weights of the network, what you are actually training is what should be the filters that the network should apply. And the thing is that we usually have hundreds of these filters for each layer, and we may have 10 layers, so then it's many weights. These are the parameters that are trainable and that we need to learn which is the best combination for that. So then the question is, how do we teach this filter network? Okay, and this, does anyone here have done any machine learning course or anything like that? Not at all, no? Okay. So the strategy we teach is very similar to, or were very much motivated back in the time on how we humans also learn, right? So when you need to prepare an exam, you usually get many examples of other exams, right? Like you do your exam at home, and then you compare with the correct answers, and then you learn how to do the exam. I don't know, you get knowledge, but you learn how to do the exam. And this is called supervised. So we have the input and the corresponding output that we would like to produce with the network. And then during the training, we show the output and we compute some error between both, and then we update the weights. And this is called supervised because we always have some example, or we always have the ground truth data that which we are expecting from the input. But there are many different ways of doing that. And it's not only supervised, we have generative approaches or unsupervised, if you're going to use it afterwards. That is like, well, I don't have localized images, or I don't have the segmentation, but I have some sort of data set of low resolution that has a specific data distribution and some sort of output. They are not the same field of views, but both sets, they are big enough as to represent some sort of distribution. And then the network learns how to translate these images into this one. Okay? And this, we can dig deeper into this later, but very briefly it's like that. Then we have self-supervised immediately. So this one, it means like, okay, I don't have two types of images. I only have my noisy data, noisy image, and I want to remove the noise. So then there are some strategies that is you degrade the image somehow. So I will remove this patch here, this patch here, this patch here. And I try to train a network that with this, in the best case, it will get this one. And because the structures in the noisy, so we have, when we have an image, noise is a random artifact. It doesn't have a structure, right? It's some kind of random intensities around the image, while the content in the image is stable, is in a structure. So when you provide many of these examples to the networks, they learn how to produce the structure without the noise, because that is what is repetitive across the entire image. And this is what is related to this part here in the upper side, and the part here, and the part here. So you use the image itself by removing some sort of information to train the network, and this is called self-supervised. So in your own, you learn this the same as when you learn yourself to know when you are feeling sick or when not, or when you are feeling happy and when not. So you have your own things and you learn that way. And then there is this semi-weekly supervised that I find it quite useful, especially when you have to annotate data. So here I'm showing you an embryo of Xenopus. And what we want is to segment each of the cells here, right? But rather than annotating each of the cells, what we produce is some sort, we just did like this with the mouse. So we produce landmarks, or the landmarks, we create circles that look more or less like the cells, but not exactly. And these can be produced very, very quickly. And then when we put it in the network, as the network is learning features in the image, the best approximation to our circles are actually the segmentation of the cells. So it will automatically learn how to segment these cells in this embryo. And this is called semi-overtly supervised. So these are approaches for you to generate data, and I think they are important with respect to the possibilities you have in the microscope and in lab. We will go with this one because, well, it's the very basic one that everyone should know, and it's the most common one, and the one that you will always make sure that what you are producing and the accuracy you are measuring is the correct. So how do we train these networks? It's very easy. We have, yeah?
I just have one question. So there are several approaches to train the network?
Yes.
And my question is, so how do you choose the best approach for your data?
Well, first, if you don't have ground truth, you cannot You cannot do this one.
Yes.
So then you need to choose between these three.
Okay.
This works for segmentation. So if you want to do denoising, then this one doesn't work. And then depending on the method, so the networks, they have names, all these deep learning methods. And depending on what you choose, it will use this approach or this one. So I'm not sure I put it in the slides, but I can show you later. So usually, no, I don't have it. So there is a schema, and usually when you have your data is what is the object, what is what I want to quantify. And you know what you want to quantify, then you say, do I have data for this or do I need to acquire? And you acquire or not. Is there any tool that does this for me, or is there any trained network that does this for me? If it's yes, super good, because then you just follow whatever they recommend you. If there's no, then you need to think if you need to train a network from scratch, and if there is a tool that with your skills and your capabilities enables you to do that. And there's one that it's the one in the practice, it's proposed learning for microscopy. But if not, if there's really nothing that can help you doing your analysis, the best way is to try to collaborate with someone, or to go to these forums, or to ask around in your institute. Sometimes in the imaging facilities, they can recommend you something, or supervisors or whatever. But that's a bit the logic.
If you had to choose between, imagine you have loads of data and you have your own groups, you will always go for the supervised one.
Usually, yes.
So in our lab we have Antonio, of their lab, and I think he used the unsupervised one to train something. And I wanted to ask, so we have loads of data, and he just trained it for a bit, and the result was very good.
Mm-hmm.
So my question is, why wouldn't he use the supervised one? Because of the amount of data that you can train the network, or?
Because I don't know the data.
Okay.
But are you sure that he has a ground truth data?
No, I think that's why he used the other one.
Okay. And what do you have?
Yeah, sorry.
Okay. Any more questions?
No?
Okay. So the way these networks are trained, it's kind of straightforward, the idea, and of course there's a lot of math. So you have your input image, you process it, and you generate a number. Okay, and you compare it with the ground truth. These two images, they are not the same. So you can compute the mean square error, for example, between them. You get a value, and you update these weights. So when you update them, the output will change until it looks like the input, like the ground truth you have. And when they are exactly the same or very similar, you stop training. So in reality, because this was a drawing, what it happens is that we have these input images and this ground truth. Here we have our network where each of the filters, the weights you are seeing here, are randomly initialized. They are complete random numbers. Okay? And it will produce something because it always produces something. And this output at the beginning, it looks super crappy. It's not like what we want. So what we do is we compute the loss. So we measure the similarity, the cross entropy, the mean square error, whatever loss function we decided to choose. We update these weights that we have here, these values, until the image is exactly the same. And when we have this situation, we say that our network is trained. So now, how do we choose which are these images and these ones from our dataset? No, sorry. Well, the way the loss function will do, what you do all this training is basically a function that decreases, so the error is smaller and smaller until it arrives to this plateau, this point where the network seems to not be learning anything else. And then the question here is, well, did it really learn anything? So how do we evaluate this? And this is when the criterion to choose the data is super, super important. So we usually have a lot of input and output images, right? We annotated our samples and whatnot. But we need to split the data, and we need to split into training and test. And there's something super important here. All of them, they belong to the same distribution, right? Like to the same cell type, to the same sampling conditions or whatever, but they need to be independent. So we cannot cheat. We can, if we were studying for an exam and we tried to study with the same exam that we got at the end, we didn't learn anything. We just learned how to answer the questions of that specific exam, right? So that's why this test data has to be very different from this training data. And independency in microscopy means different field of use, different replicas, and any sort of variability that you identify from your experiment. Okay, so if you want to identify between sick and healthy patients or samples, you have to have both here and both here, but the patients need to be unrelated. Okay, and then what we do also during the training, we extract a very, very small set from the training data to validate what we are training, whether the network is really learning or not. And this loss function for this validation data, it usually looks like this while we are training. So we don't pay much attention to how the loss function looks in the training data, except the fact that it's learning and is decreasing, but we really focus on this validation data to check that it not only does well for the training, but also for the validation, for the data that is not used to update those weights. And then is the question of overfitting. So probably you heard a lot about all these networks that learn very well in the data, but then they don't work very well. That's called general stability. So how general is my method? And in numbers, what it means is, if I have a function that it needs to be adjusted to all these points, and I have many, many, many degrees of freedom, then I can get the perfect polynomial, right? But if I give a new point here, this doesn't fit in this polynomial. So this method is not general. It's just very, very well fitted to whatever was given in the training, but with new data, it doesn't do what it has to do. What we want in reality is a function that is able to gather the pattern that is intrinsic to the data, and not a function that is super perfect in each of the points. So that in average, it works well. And then you will say, okay, Estibaliz, what if I'm in this situation? You need to hire a model. Okay, there's no other solution. So you really need to sample properly the space, and this is the reason. So if we have very few points, because our networks, they have many parameters, they will fit very, very well to the training data, but not to new points. If we have many samples, then we will get a function that does very well on average. So then it really identifies the pattern that we have experimentally, because our data is not exact. I mean, there's always noise, there's always artifacts and biases. And now the question is, okay, Esti, but I cannot have this in my microscope because I cannot be a part of 3 years. So how do I move from here to here? How do you think you could create some data from here that looks like this one?
You introduce some the reflections of your
And how do you do that?
information.
You don't know? So imagine that I have this point. How can I generate a new point from this one?
You split into like an
I move it a little bit and I give it the same value, more or less. And then this one, how can I create a new point? That is similar but not the same.
a different transformation.
A different transformation, right? So I can move it a little bit to the left, maybe I can even move it down. I mean, it can have some noise, of course. And that is how I sample new points from my data. That is called data orientation. And with images, how can you do that?
Subtraction.
Subtraction? I mean, here we just move it to the right and to the left, right?
Yeah.
So what can we do with the images?
Get rotations.
Rotations, shifts. And I have noise?
Yes.
I know. Right? So all those sort of transformations, they work. So here, for example, what we did some rotations, and it doesn't really matter how I do the rotation, this is a cut, and it will always be a cut, and a cell will always be a cell. And actually, it's not only a cut, it's always the same cut. Okay, so if this is Benito, my cut, it will always be Benito, and I can teach that to the network no matter how I change the cut. The same happens here. So I can remove parts of my image, and this will still be a box, and this is a plane, right? And then here we have an example. This is the number 6, and it doesn't matter its orientation. It's the same with cells. It will always be the number 6. So all these are the sort of transformations we can apply to the data in general in computer vision. But do you think this transformation will work in microscopy?
No.
Why?
Because you're out the contrast?
No?
No, you have no colors.
You have no colors, but I could add colors, no?
But that doesn't change the pixel of thing.
With noise, you also change the pixel value. If we are processing channels, channels in microscopy, they have a meaning, right? They have different structures, so we cannot change colors in the sense that we cannot decide to put in the red channel whatever is in the blue, and so on and so on. So in microscopy, there are some things to consider. First, that the transformations will apply to the image, it should also be applied to the ground truth, and that we need to preserve this biophysical meaning. And this means here you are seeing some extracellular vesicles, and here what I did was to rotate, but I rotated that much that now what I have here, it makes no sense, and here I have half of my exosome. So if I go to the network segment this, it's going to tell me, well, I'm not seeing it completely. So this rotation was too big. We can apply, but we need to be careful. If I decide to do zooming, you do zooming, but you preserve some sort of the original size that you expect in your sample. And also if you do some shearing, well, this is not an exosome or an extracellular vesicle. Okay, so they are rounded always, so this is not real. And then again, we can add noise, we can add contrast, but we should preserve more or less the quality we expect. Okay, and so this is important. And then still, okay, we have the augmentation, we have all these things, but still we need some real data. And this is not always possible. And you will see, you cannot be annotating for your entire PhD, and you cannot be acquiring images for your entire PhD or postdoc or master thesis or whatever. So then there are two choices here. Do you know, have you heard about this?
You use previous images?
No, well, yeah, if somebody has near images to yours, yeah, that would be very nice. That's why you should always share the data from your papers. What else? Okay, if myself, I'm working in the same thing as you, and I have a model that's very, and like we have the same sort of experiments, and I have a model that is already segmented, for example, nuclear, let's say we want to augment them. Would it make sense that you take my model for your problem? Or would it make sense that you annotate 100 image row? And so on.
I will take your model.
You take my model, right? So that, and the model maybe is not perfect for your data because our experiments, they may look different because of the human variability. So then what would you do with the model that knows already how to segment nuclear models? Modify it slightly. And that's fascinating. Okay? And it's called tuning, and then that's what I mean. So the very nice thing about models is like once they are trained, they already know how to extract some features from the image. So sometimes we don't need to have hundreds of images if the task we want to perform is the same, but the data just changes slightly. So this is the the anatomical part of Arabidopsis is on one side and this is on the other side. So it already knows how to identify the edges here. But just for the new data, I would just need to train it slightly. So this is fine tuning. Okay, so it's basically for the same task, but when the data changes slightly. And then there's transfer learning. This is a little bit more abstract. So usually when we have these networks, the networks have two parts, the encoding and the report. The encoding side of the network is at the beginning, they take the image and they compute many features and they make them very small, but in vectors. And this encoding, it usually already knows what is the spatial distribution of the images, more or less on features, right? So if we train it, let's say this network that it gives you just one number for the input, this was Benito, it already knows that Benito has eyes and a mouth and 4 legs and some ears. If now I want to segment Benito, well, it already knows how to identify Benito. Right? So it knows some sort of features there. So I could take this part here and just get an output that knows how to decodify these features that contain information about the ears and whatnot and use it for segmentation. And this is called transfer learning. So it's when you get some features from a model that already knows doing a task for which it needs some features same as yours, and you take it into your model. This is you are not gonna do much into this unless you go into programming, but you will hear about it. And these are the main two things. And well, just because, of course, I need to mention, so now there are more and more initiatives to put these models available for all of you. Yeah.
I have a question. So in the fine tuning, do we use the features of the model that we got, or do we train these features to our?
No, so you take the model that was trained already.
Use those features, you don't change them.
Yes, you train it a little bit more, but in this loop of updating weights, rather than updating it 1,000 times, maybe you just need to update it 10 times.
With our data.
Just a little bit.
With our data.
With your data. Yeah, with the new data. Yeah. So this is important for many reasons, not only to not be taking images as crazy people, but also because training models, it's really a hard task and it's also quite polluting in terms of the CO2 that we are producing and so on. So there are more initiatives, and one of them is the BioMed's model soup, where Ivan and I are working, that is to put all these models available for you, so you just need to download and fine-tune them a little bit, or even use it directly with your data, and so you don't struggle doing things from scratch. So then, very brightly, we have these 4 components of a model. So the model architecture is how we combine these convolutions, and usually you will hear about UNet and many other sort of technical stuff that you just need to know that this belongs to how the convolutions are connected to each other. The loss function that we use, depending on whether we just want to do a denoising or we want to classify pixels and use mean square or cross entropy or so on. The optimization I will enter on this one unless you have questions. And the data, that is how we annotate the data to train the network. So all these, they affect to the performance you have in a method. For example, if you use a mean square error rather than a cross entropy to train a model that does segmentation, it won't work well because this error is just comparing intensity values of the output, while the cross entropy is forcing the pixels to be classified and in a probability. Okay, so this is for you to have it here back in your brain, okay, for the future. You don't need to struggle too much, just know that all these may affect the inference, but it also affects the configuration. So this means that it's not the same analyzing 3-dimensional data and doing classification, then analyzing time lapse and so on and so on. So this is part of the process. When you have your images and you know the task you want to do, then you will need to go into one architecture or another one. So I don't know if you have questions until here. So I hope I communicated that this is not easy, okay? And it's not a quick fix for your data, for your analysis, because you need to create the data, you need to evaluate it, you need to maybe annotate it manually, means drawing, literally, many images. It means evaluating all the architectures you tried. It means knowing how to train your network. It means GPU. It means getting your results and make sure that it will work with new data. So yeah, it's very powerful, it's very good, but it shouldn't be your first choice. Okay? And I hope I convinced you about this, unless a real expert comes and tells you you cannot solve this with anything else. Okay? So always going to teach you or something to play around before going into deep learning. So I'm going to show you some examples of why this can be very, very tricky, or what you should consider. So I spoke before about getting a very clear idea of what you want to do with your image analysis. And the thing is that if you want to track cells, as we saw with the bacteria, we have very, very great segmentation. But that doesn't ensure that our tracking is going to be good, as you saw it already. Okay? So then I told you maybe what you want to do is not the tracking. Maybe what you wanted to do is just count the number of bacteria across time, and that's it. So that is the first objective. Here I told you, if you want to measure the morphology of these mitochondria, then this circle is not good. But if you want to measure the fluorescence intensity of these mitochondria, this circle is perfect, because if you average the fluorescence intensity in this area, it's going to be very, very similar to the one that your mitochondria has. So then that solution is good enough and is your objective. There's one example that I found it quite funny. So Patrick Miller is a researcher in Google, and they did some work. I mean, basically some researchers hired them to get a model that was able to do some diabetic retinopathy diagnosis from the eyes, from these scans that they do it when they go to the doctor. And basically what they wanted is to speed up the process of identifying when the patient was actually needing to go into some kind of treatment. And they tried to train a network to identify whether the scans or the patients they were seeing were actually suffering from this specific disease. And the problem is that when they were generating the training data, the doctors, they were not agreeing with each other. And this is a very common thing in medicine, especially when you do diagnosis. And the worst thing is that the correct answer, if there is any, sometimes is not the most common answer. It's not the answer that most of the doctors gave you. So then what they realize is, okay, we don't really want to know whether the patient has this specific type of diabetes. We just want to know if it needs to see an specialist. So then they change the approach, and then they train the network to say, okay, this patient needs to go and see a doctor. And things work, because in that kind of question, most of the doctors will agree. While in the other one, they won't agree, I think, on average level. Okay, so the way you ask and you formulate your questions is super, super important. So then another mistake that is very common is to try to see things that they don't exist. So this was my PhD project. I wanted to see how cells were using cellular protrusions to grow in three-dimensional space. These were cancer cells. And the research that was done until that moment was that to say that the cells, they have the body, then they have the model protrusion and the branching structure. So then our collaborators, they told us that it would be super cool to have a model that was able to identify those branches. So yeah, that's what I annotated for 2 years. I got my accuracy metrics super cool, all of them. And then when I was analyzing your data, things didn't work. Why do you think they didn't work? I mean, the network was completely, we didn't know it.
Error? Error?
But do you think it is valuable?
Did you overspeak everything?
I was trying to go from here into here, and it was filled with biologists. Do you think that's correct? The cells have any sort of backbone or anything that distinguish this structure from this structure in reality? No, it doesn't exist. So this objective is completely wrong, is wrongly formulated. If I try to define where's the line between the cell body and the protrusion, that line doesn't exist. So a network, of course, will never learn to do something that does not exist. Okay. And this was a very basic formulation, and it was a very basic mistake. And it was very clear to the biologist. So you should never ask a computer to do something that you are not able to see. And magic, it doesn't exist. Okay? And AI is not magic. Okay? So this should be very clear. So then, well, we changed the approach and we said, okay, segment, you can do it. Yes, tips are very well defined and point of the thing. So then we can just go into a different direction, try to segment the cells, get the tips, and then track and get the analysis. Then another problem was that we wanted to identify the cells in mitosis in this field of view. And then I knew that statistic was a variable that we were using, worked very, very well most of the time. But when I was training my network and putting this data into the network wasn't learning absolutely anything, and everything was empty. I didn't have any cells segmented. And the problem was that the network, the only thing that it was seeing at the beginning was this patch. And I was trying to tell it that this thing that it was seeing was the cell part. And as you can imagine, we don't see anything that is telling us that this belongs to a cell, right? I mean, this could be the background. So, getting back, the problem here was that my colleagues, that they love super resolution, they were using an objective of 63x image cells in phase contrast, which is like, no, because you are using almost 10,000 pixels for this field of view, right? So then each of your nuclei, I mean, you don't need that many pixels to display these cells. So this whole square that I'm showing to you is 500 times 12. This one is what the network is seeing to determine whether this pixel belongs to a cell or not. So in this case, yeah, the size in pixels was super good, resolution was not bad, but we didn't need this resolution. And this is a way to see it because it's a bit technical. It's like when you train networks, you give them some input that is usually crop, right? It's crops of 500 times 500 or 252 times 252, and they see that to learn. So you can display that and say, okay, this makes no sense. But another way to understand it is if you like going to museums, they always have the place where you sit down in front of a piece of art at a very specific distance. And that distance is the correct one for you to appreciate the art you are looking at. So, in other words, here, I'm showing you some drawings from Monet. Do you know what is here? What we have here? Does anyone of you is able to see we have that?
Like entrance to a forest or a path?
And the forest or a house.
Yes.
Forest. Yeah. And here? Right. So here these are supposed to be the house here on the forest, and these are supposed to be flowers, and in the background a house. So you need perspective, and if you want the networks to give you some outputs, understanding the context, you also need to know what the network seeing to classify the pieces. And this has to do with the resolution, and it happens many times. So these are all the drawings. So if we just want to segment the square, our images need to be much smaller than if we want to segment or we see each of the patterns in the pieces of art, right? So this is very similar, and I hope with this is more intuitive for all of you sharing the network. So yeah, we downsampled the images. It was very simple by a factor of 8. So 8 times smaller, and then things work quite nicely. So yeah, so now I will just very briefly. Yeah.
I'm not sure if I got the final conclusion. Right. You were saying that we should be careful about the resolution of the data that we use to train our network.
Yes. Yeah.
Not to use a higher resolution, that's what it needed to address the problem.
Yeah, so high resolution is not always the resolution. So yeah, what I'm saying is like when you train your networks, and you will see it in the notebooks, you need to decide what is the input size of the network, right? Like what is the image you're giving to it. When you display, you should always display that. So in this case, it was this image. And the problem that we have is that if you take 1 pixel of this image, sorry, the input was this one, sorry. So the network was seeing just this to segment this. So it's not, I mean, there are no features here, there are no edges, there are no things that tell you that this is part of a cell.
Yeah.
So then the network is like, well, you're giving me random black and white patches from things where sometimes I get some edges if it got here, the drop, but I'm not able to compute anything because I don't see anything, right? So you always need, and these perspective things that we need to see things from far from is exactly the same for the networks, and that's why it's called computer vision. Okay, so it has to do on how our algorithms see the information. So this being said, well, yeah, the learning in microscopy is a super hot topic because it gives us amazing results for quantification, detection, denoising, but something that is super cool, I think, and it's really, I mean, I think it's something that established a new kind of era in microscopy, is that we can start using these artificial kind of labelings and these super resolutions to start building smart microscopes. And that is what we do in the lab. Okay? So basically, we use the fact that we can detect in real-time events to decide when to image something. We use the fact that we can track some sort of activity to switch between confocal and state, and so on and so on. So this is why it's actually becoming super interesting for us. And then this is very cool, but then I want to ask you, why do we want to have these super cool microscopes?
Just have the data
But imagine that I'm a super rich PI.
Okay.
I can hire 20,000 people to do experiments 24 hours. And I don't need that. For a fish? No, I don't care. I think Hannah said something related to this. What's one of the major limitations of life cell microscopy?
Phototoxicity.
Phototoxicity. Why?
Cells are hard to keep happy.
But I don't care about cells. They don't shout at me. I don't want to have them happy. They need to all the skills. my image, then I don't care if they're happy or not.
If they're not happy, they
But
want to
you
behave
stop
in the
with
way
the
that
They want
you
to
want
behave
them to be.
in their way, and that's
Physiological behavior or.
exactly. So it's not physiologically relevant if I'm altering the health of the cells. So for the toxicity, what it results in the damage of different components of the cell or DNA. So then the reliability of my experiments and even the reproducibility is completely broken. So so then I will show you a video. This was one of the very first works in deep learning for restoration. And I think after this, maybe I don't need to tell you much more. So this is an embryo that is imaged with a very high laser power. It's moving a lot. Doesn't like it, because when you reduce the laser power, it's relaxed. It's happy, seems, I don't know. And then, yeah, the quality is lower, but if I'm able to restore it with some deep learning, then I get much better quantification. I can track these nuclei that here I couldn't, and also this is a more natural behavior than the previous one. So that's why it's super interesting. And then, so then the question is, how can we play with this, right? So because you can say maybe, yes, we need fluorescence. So something we are doing in the lab is like, because we have these networks that are able to translate phase contrast into fluorescence out of noise, then what we can do is our live experiments using this lower quality kind of imaging, translate it with some kind of artificial labeling, and then doing the segmentation and tracking here without having irradiated ourselves with any sort of light. So this is what you are going to do today. So actually, I will give you these and these images, and you are going to get artificially this image with your computer, and then we are going to segment it. So, of course, there are many more strategies, and again, here, not advertising at all. We just got out a perspective that is discussing what are the different kind of approaches you can combine virtually with your acquisition to try to reduce the laser power or the number of times you do some imaging, and also how you can work with fixed samples to create the training data and then use your method with some live imaging data. And then, because I'm going to share the slides with you, this is all the sort of open source and user-friendly tools that you have to use deep learning with your own images. So you don't need to know how to code anything of what I explained to you today. And of course, there are some games you can play online, and some comics you can read. So Pocket Flame is a very, very nice blog in biology to search for new tools and methods for you to do some analysis, and they do very nice reviews, and I think it's a very, very good place to go. Of course, the images that I told you. And then probably you heard already about this sound, right? Who didn't hear about it? Did you read it? From Facebook. I don't know, it's a news event. No, this model that you upload an image, you just click with your mouse what you want to segment, and it will automatically segment everything. No? Okay, so this is a very new development from Facebook that was basically very smart intelligence already there. And there's already some plugin in Arrate and so on that you can use. So you label your cells where they are, and it will segment it to you in an interactive way, and it works in a high percentage of time nicely in case we don't want to train it. So with this, I would like to thank the lab, of course, the Carbon Ticket Lab, and the collaborators with whom I work in some of the things I showed you today. We can go to the second part. So for this, if you think in that part, the idea today is that we are going to train a network that does this. And then we are going to take this and we are going to use PG to segment it. Okay? And then you can repeat this experiment in your lab whenever you need to segment or track cells. You can just, if you go with phase contrast, has anyone gave you your phase contrast directly in their research? this concept, right? So you can do exactly this sort of experiment. You just need to prepare some samples, to ladder them, to get the training data, and this is only 2 days of research, not 3 years of annotation, and then yeah, you can find. So I will show you how to dig into. So if you go to the PDF file, here, well, it explains you the type of data you're seeing. So basically, we have Chinese hamster ovary cells. That's what Pedro was the one preparing this sample, so you can ask him how much time it took him to acquire the data. They have some labeling for the membrane, so then it's very easy to segment the cells with some method. And what we need to do is download the data. You already have it. So go to this GitHub. And this is 0-point deep learning for microscopy. So what they do is they provide a platform with some Jupyter notebooks. There are some sort of notebooks for Python, but they hide the code, so you don't really need to code, but just give the input paths and parameters that they tell you. And they have many different deep learning models that you can train for different image processing tasks like segmentation, denoising, restoration, or whatever. And what they allow is to connect with Google and your Drive and some cloud computing services for a very limited amount of time. But I think it's enough to start playing with this. So here it says you go to the Pix2Pix. So this is the whole wiki with tutorials and whatnot. It will be on Pix2Pix. They explain here how it works. So it's a conditional generative adversarial network. So it generates fixed images based on some input structure. And then it has some sort of agent that is telling them that what you generated is false or is true, and that's how it does. So we can click on here, open and pull up. So this is the zero cost, the GitHub, or just only from it. I can click on each of these and then open the code. So before anything, have you logged in your user in Google? Okay, so now go to your Drive and upload the data that you have in the folder. Pix2Pix, yes. Yeah. Okay, but we can run the rest. So while it's uploading, you can get back to the notebook. Okay, and here in File, you need to say save a copy in Drive. Because the notebook you open, it's not stored in your Drive, so you really want to make sure that you have the notebook there. And then a new link will open. In notebook, if you go to your Drive, you will see that now there's a new folder called Collab Notebooks. And here you will see the copy of Pix2Pix. Okay, that one. So now I have it already open. Yes, but didn't it open automatically when you click on save in my drive? No, it saves automatically in your drive. So if you click save a copy in my drive, I think yes. In the same time. Is this one?
Sorry.
So did you click on open column? Yeah. They have to be right. Okay. Yeah, sure.
Please upload the data to your drive.
The text and print.
Yeah, all the things can be created.
If you don't have such
Yeah, in the report there are the requirements by somebody who wants to run. Yeah. Or it's connected with a pseudo or any sort of association. Yeah. So besides you, is there anyone here that knows Python? So now, we are going to make sure that we are using the GPU. So we go to running environment. So I don't know how it's called here in Portuguese or in English. And then manage change type of execution, yeah, whatever. And then make sure that you have this GPU one. I mean, if you're not going to use a GPU, it's better if you have a CPU, because otherwise Google will cut this service after 30 minutes or 20 or even before, okay? So make sure that you have this. If you have it already, then just cancel, if not change it, and save the changes. So now, this is a Jupyter notebook with code for Python, but the code is hidden, okay? So if you have text, you have cells, so this is a cell, and this is text. So if you double click, you see how the text was written. And we also have code like here, okay? And these cells that have code, they can run. We can just click on this, play, and they will start running. So then the idea now is that you run the very first installation. So please click this one as soon as possible because it takes some time. And it will start installing the dependencies that we need for this notebook and this network and this sort of algorithm in a virtual machine in Google Colab. Okay? So it's not in your computer. If you disconnect your laptop from internet, this will be disconnected. So now we click this one, and we can also click here to check if the GPU is running. We can start with some of them, yeah, and to create the result. And then it will ask you to give access to your Google Drive. So basically what you're doing is giving Google access to check your personal data. It's up to you how happy or not you are with that. But we need to have some sort of storage things in Google with this here. Yeah. I guess if you have an Android phone, you shouldn't be very worried about this. To be honest with you. Yeah, I will just say here connect. Okay, so I connect. And there, yeah. So you have the access here, running environment, and then change running time. And a pop-up window like this working. Yeah, and then you need to it's right now free. Yeah. So now if you connected already with your drive, you will see here in the right you have access to your Google Drive here. So my data, so try to find where you have the Pix2Pix data. My data is here. So now we need to start giving the parameters. So we need to tell, okay, what is the folder containing the training source images? So you can say, okay, this one, you click on the 3 points and you say copy path. So here is my folder, and I can say copy path to this folder. And that is what I paste here. And the same for the membrane. And then our data is grayscale, so it only has one channel. Are you all following? What? So we click on the 3 here of the so we are gonna train the network, right? And we need input images and the ground truth. So we need to tell to the code where are the input images and where is the ground truth. So the input images is the pH, that is the phase contrast images, and the ground truth is the membrane, is what we want to train. So training source is phase contrast. So you need to provide the path in your Google Drive to phase contrast. And the target is the membrane.
The problem is that the folder doesn't appear.
Just click on this one here, refresh.
Yeah, I did.
But is your data?
Yeah.
Now, please refresh. But which one of your it's all
other name on the back is then save it in a way to do Yeah.
So then besides the data, you need to choose what's the name for your model. So no, please grab instruction. If you decide to go on ahead from me, then we shall be okay? Yes. Yeah. Or just well, actually, you can run 100 textbooks and wait until 6 or something, and then you show us your results. But please read the instructions if you decide to do it in your own. Okay. Okay, so here we'll just write the date for today. This is the name of my model. This is where I want to store my model, and make sure that you have a path in your drive, because otherwise you will lose all the things you generated during the training. And then for the epochs, please write here 30. Don't write more. Okay. And then this is the batch size. So this is the size that we crop in the image on each iteration of our training. Okay, so because the entire image is very big, we try to give that to the network. We don't have enough memory, so we usually crop little patches, right? And that is what we give to the image. So you can choose this size, you can choose a smaller size if you want. This is the batch size, and this is the learning rate. So the learning rate, the smaller it is, the slowlier we are gonna learn. So basically what we are trying is to get some weights, train it. If we have a very big learning rate, on each epoch, we are updating the value of these weights. If we have a very big learning rate, the update is gonna be very, very, very big. So then what happens is that if the network goes closer to the output that we expect, but we have a very big learning rate, in the next iteration it's going to change a lot. So then we are not going to allow it to slowly get optimized to the output that we want. So usually we write something that is not very big, but also something that is not extremely small, because it really needs to have some sort of space to move, okay? So I think this one is good. So once you have it, you can run it, and it will show you the input and output images. Okay, an example. My training was finished, and I finished with a quality check. So so what I told is like, yes, I want to evaluate this model, but if your connection again was broken or something, and you already run the training and you just want to evaluate the model, you can just unselect this one and provide the path to the model you want to evaluate.
Reviewing that?
Okay.
Do
you want to assess the model we just trained it? Okay.
It's just.
Yeah. So then we are going to run the quality control. So we are going to test whether really the model learned anything and it's doing correctly. So what is the data we need to use, the training or the test? Is it clear for everyone? Sure?
Exactly.
So then we write the path to the test. And then here it says, do you want to prepare the data? This is some processing that I need to do to give it to the code. So because it's the first time we're doing it, yes, we need to prepare the data. If not, because this data is prepared, I mean, here there are 3 images, but when there are 100, it takes quite a long time. So if we already did it, we don't want to do it again. And that's why it's asking you. Then the normalization of the input and the output image, it should be the same one you chose for the training. So it's the one by default. And then here, imagine that you train your model for 1000 epochs. What this quality check is going to do, because this is not always like that, but it's the case to fix. It takes every epoch that is stored and it will process all the test image and save it and then produce some value. So if you have 1000 epochs and 1000 checkpoints, you don't want to evaluate all of them, but maybe every 20 or every 10 or whatever. So that is the value, but here, because we train very few steps, I just chose one and you should choose one to see it. Then there was the very next question, that is the patch size. So when we train the networks, we crop patches because we don't have unlimited memory. So we usually crop like a patch around the image, and we do it in many different places of the images. And then we introduce this in the network to update the weights. But then when we do inference, because we don't need to run that many parameters, we can give as an input to the network a larger image to process. And this is the size that is asked you. So usually you can leave it like that. If the image is smaller, it will drop it. But this is important in the case where you have images that are super huge, because of course, I mean, those, they will really penalize the memory, right? So this one you can leave it as it is. So I run it, and I run the quality control. So what it's doing is to process with each checkpoint of the model the test data. So if I go to my model, this is the folder that the node created in my drive. So these are all the checkpoints, all the trained models at each epoch. Okay, it's a file for Python for the code. And then we also have the PDF file. And this is super important, because this one is telling you what was the configuration you used to train your network. So in my case, it was this one, but some of you changed the learning rate, some of you changed the batch size, and that is okay. I mean, when you use these networks, you need to run several trainings until you make sure that you got the proper training, and there's not one correct solution. Okay, you need to do some kind of parameter search. And then, it shows you an example. So this is like a report that is very nice to attach to your publications. For example, if somebody needs to reproduce your work. Then this text file is for the training and loss, so you don't need to check it. And then when you run the quality control, it creates a new folder and it creates a new report. So this is the value of my loss for the test data at each epoch. This is an image with the quality check. That's what we will go through it now. That's just really like that. So then this one, yeah. And this one in the report, you can see for each epoch, okay, what were the quality measures. In this case, we're doing virtual labeling, so we are not measuring any kind of Jacquard or true positives, but we are measuring a structure similarity index and this kind of measures more for a kind of regression models or models where we are not classifying pixels. And it tells us each epoch what was the values. And again, this is for you to report your work. Okay. So now, if we go back to the code, when it finishes, you can see here the two curves. So you can actually see the SSC got increased upon time, so the images are better after some epochs, the output. And the loss, well, this is not the loss, this is the LFX, is another accuracy metric that measures how realistic the image looks like compared to the ground truth data, and it does some sort of things that I will really not go into that because it uses some retrieval networks and things, but it's kind of a perceptual metric that it measures how realistic the output is. And this is an error, and it got smaller upon time, so this is okay. Are your training curves looking like this?
Not equal, but it reaches the bottom.
Anyone wants to show here? Who ran it for more than 30 epochs? There, guys? 30? Okay, got it. So, I will take one of your laptops at some point, even if you don't want. Okay. So here it asks you to choose one image to show, okay, and which epoch. So I will take the latest one, and we'll show it here. It will be updated in a second. So, this is the output of the network. Does it look good to you? These are the inputs. And these are the differences between both. Okay? So I think it's pretty cool, to be honest. But I think yours should look much worse than mine. Because I use an internet model and already knew how to do. So if we choose, for example, can I take one of your laptops? If it is so errors, when things are better, they got smaller. Yeah. But similarity metrics, they got higher. The more similar, the better. So the first plot, got higher because the output of the network and the ground truth, they look more and more similar. And then in the other one is decreasing because the error is smaller between them. I would ask if it was something that you would accumulate during class on that specific point, right? Yeah, so the thing is that the network with the epochs is learning something. So what you would expect is that it's decreasing upon time because the situation of the network at this time point, it depends on the previous one because it was updated with the previous weights. So, yeah. So, who has a laptop with an HDMI entrance there?
So there, and so mine started increasing here. Hers started increasing here much later. And one of the reasons is because I used a model that was already capable of translating images into fluorescence. So that's why it got much later. So now if you go to your results here, I don't know if you remember my images, but they were slightly different, no? Yeah. What's the problem here? So this checkpoint is this warning is the number 5. So it's at the very beginning with when the network started learning something. So what do you see here? For example, this one. If you, it looks blurry there, right? Yes. And so this is a cell that probably, I don't know, if it was dividing or something, and they were very close to each other. So at the beginning, it was not able to compute the difference between those. But if you take the latest checkpoint, it will update at some point. It takes some time, but it should update. Unless it's below. No? Why is not changing?
That's fine.
Pretty much the same.
It's not changing, no? Okay, so below, can you check below? If it computed something? Here? Yes. Go down. Now, there you have it. So that is the same image, but after 30 epochs, so you can see that it starts, drawing those lines. So it starts to become more specific or specialist. And if you go down, you should have the other image. Down. Yeah. So here, this is another checkpoint. And for example, something that it's kind of clear is that this cell, if you try to get the cytoplasm in the left, here, you almost don't see it. And one of the reasons is because from here, it's very difficult to infer it. So you really need to train many epochs. So this is something we did it super quickly. And there are more. I think these two brightest spots, that they have this very clear artifact here, it knows that there is some fluorescence, but it's still, it doesn't know how to do it more realistically, and so on and so on. So, yeah, I think that's it. So now one thing you can do, I think it's okay. Thank you. So one thing you can do, if you want, is to leave one network training while we do the rest. If you want to check what happens after many epochs, or I can show you something. It works super well. The output is really amazing, but you saw that it's already working very well. So, yes. So, if you go to the folder in your drive, you will see that in the quality control, you have different folders with the outputs. So you can download them. So we can open in Fiji and check how so
you can use the numbers, those are the output at each epoch. So you download one of them, ideally the one with the best quality is the latest, but you can download both and see it out. So in my case, the first time I trained the model, this was the output after 5 epochs. Okay, and I think, it's true that it's not perfect, but I think it's quite good. Good segment starts from here with the limit. Okay, it looks quite nice, not very realistic. This is the ground truth. So then So then after, I think this was 20 epochs or 30, it already looked like this, which I think is very, very nice, but it's still, you have some nuclei like these ones that is not able to infer correctly, and some cytoplasm that still doesn't have a lot of contrast. And then, from here, today I trained 30 more epochs, and I got this one. So, from here to here, it didn't change much. be honest. I mean, here, a little bit of limit, but you can see that it starts converging. Yeah. Okay, so here there are some things different, but it's quite good. So now the thing is like, okay, can we really use this for segmentation? So now, I don't know, maybe I give you 10 minutes to play in Fiji with the images you just created to see them, and then go to the next for the segmentation. Okay. One thing you can do is to overlap channels, for example. They can hide the fake ones. Yes, 1 second. What do you think about your results? Would you use these results for segmentation? For morphological studies? And phenotyping? Why not?
Because the result that you have the algorithm has difficulties in segments that are very large.
We didn't segment out anything yet.
Ah, okay, sorry. But we don't really like yeah, this one.
This is your last epoch?
This is to
No. Is the result of the last epoch of your algorithm?
no.
So you usually choose the results from the most accurate. So when you look at the graph here,
Yes.
at the graph up to up. No. So you choose the one with the highest accuracy, no? Okay. Why did you use the lar and you go to the other side? Now enjoying the training. Yeah. After. Yes. So this is the most accurate point, no? This is the one with the lowest error. So then you take the results of the latest. Okay, so my results were these ones, and I think they are pretty cool. So imagine that you have this. Would you use this for self-phenotyping? For segmentation? I know you have something in your interior that tells you no, because it's not real, but why not? Yeah, so you trust more Pedro than my algorithm, right?
I didn't hear it was
It's a very consistent. So, Pedro, you can get out of the room, and then so, the information we are inferring in the images come from a real image. We are translating it. And fluorescence is not giving you the, the fluorescence we infer, we are going to use it to delineate cells. So why couldn't I use this even if it is fake? Or what would be a way to make sure yourself that you can use it? No. We are not going to use this for segmentation, for morphological analysis. So we are going to segment the images. And that's why we did this, because we don't want to annotate images. And because segmenting based contrast images is a pain in the ass. What can you do to make sure that you can trust the results of the segmentation once we segment them? Do ones by hand. Okay. But we don't need to annotate 1,000. just few of them for the test. Now, can we use these intensity values for measuring some cell activity? What?
Well, can you please say it?
How are we computing this intensity value? From a static image, right? From a fixed sample. So what is fluorescence telling me? It's molecular activity, right? Somehow it's proteins. So the network didn't see these cells, how they were behaving. So it's giving us some shape information that is embedded in the image with just a different contrast. So that's where we can use it for shape analysis. You shouldn't use it for molecular analysis. Because the intensity is fake. The shape is blue. Okay? And this, so this is the part of being critical with AI. You need to very clearly identify yourself, what you are creating that is fake, and what you are creating in a way that is fake, right? So molecular activity, if there was a time lapse, then I wouldn't say this, but there's no time lapse. The network doesn't know in which direction the cells were moving. So how the network can know what activity is in the membrane? There's no way to know that. Okay, so that is fake. Morphological information is in the image, and that is what the network is outputting in a different way, and you can trust it as long as you measure the accuracy values. Okay. So now we are going to open the terminals and write some code. We are going to use PG, but we need to do some installation of cell code. So cell code is one of these super cool algorithms that you can just go to the website. You can drag and drop here a PNG file, and it will submit it for you. You can try it later at home if you want with your data. But we can also try with one of the images. Yeah, and it will give us the prediction, and you can play around. So there's a plugin in Fiji to use some pre-trained models on cell boards. There's also a notebook if you want to train it with your data in zero cost. So if you get back to the PDF file, it gives you some instructions. So here it's a master class about Python. So to program in Python, we install Conda, and Conda comes from Anaconda. So Python is a snake, and Anaconda is another type of snake. Right? So I don't know why the name is like that. But Python is a kind of is a programming language that it's calling to C++ code, but it works with packages, so we can do 1 + 1, but we cannot say segment. If we want to do segment in Python, we need to download a package and a library. And Anaconda, it's kind of a wrapper that will do all the installations we need in our computer in order to have environment, so we don't destroy versions that we have of our code and these sort of things. So when we install Python, we don't install Python, we usually install Anaconda. And your computers, these ones, they have Anaconda installed. But if you want to install it in your laptop, you need to go to Anaconda webpage, download, and click on install, and it's very easy. So then, if you open your terminal in your Mac, and you click Conda, Conda should be installed. You should get something like this. So they install it really. So the thing here is now for your future, because once you start, we won't stop using Python. So the thing is that because when we program, we need to use packages, and packages they have versions. So rather than installing all that directly, as you do with MATLAB, what we do with Python is we create environments. So it's like creating users for your coding. So then we are going to create one environment now for cell post. We activate, and then whatever we install in our computer for this work that we are going to do now, we need to make sure that it's inside this environment, this post, this cell post box. And this way, if somebody else is using my laptop, or I'm using I am also doing some programming for another thing where I have another environment, they won't interfere between them. So I won't have version compatibility. Okay? So this is the Python environment idea. So if you click on that, you have this, and then in the instructions, it says, contact create. This is N for new cell post. So this is what you need to type here, contact create -N cell post. And when you create it, you will see something. Okay, I already get it. But it will ask you to download some packages. I will just that will start doing this sort of explanation. And it will ask you want to install these packages. You say yes. Right? Enter. And now it tells you, okay, if you want to work with this, you need to activate. Did you all go to this step? Yes. Okay, did you all manage to create the environment and you have this in your terminal? Please don't go ahead because you said you didn't know Python. So if you broke up, it may not work at all. So now what we basically do is write this. And now, before anything else, you always need to make sure that you are working on your environment. Because if you are not, and you install a version out of this box, then you are going to break any Python installation. You can't break any Python installation or version compatibility of your machine. So you then need to uninstall or come back and install it again. And well, if you have never programmed, it's okay. But if you have something working, you really don't want to go into that. Okay? Yes, you may lose a lot of things. So just make sure that this one is activated. So now it says this one. Now, so you can run it, and it says Python mode. So just try to install. I think. Yeah, just run to install the post. Okay. No. We do it after the activation. Yes, you activate. Please, who didn't activate the environment? Who doesn't have this post in the terminal? You all have this?
We have to write for that.
So you didn't have it. That's fine.
Yes, okay, for now. So now, we are gonna move into PG. So the thing is that now PG is gonna go to this environment that you just set up in your terminal, and it's gonna run that in the backend, so you don't see anything, right, in the backend. So more than in your how do we do this? So when you install the environment at the very beginning, you should see a line that says environment location, and in your case you are with Linux, so it would say like home user mini conda self post. So get this one in hand because we need to use it, okay? So now when you go to PG, open PG, and in plugins, so these people from BioP, they are people from the imaging facility of the EPFL in Switzerland, and they develop many plugins for PG and many things for the users, and they are all user friendly. So when you start this BioP, they have many other things and a lot of documentation. The last is for registration, for example. I didn't use any of them, to be honest, but these are there, and I can tell you that this team they do very good development. So if you need to do any analysis that that you find here in their plugin, I would be very okay. In this week, I think it's great. So in this case, they did a wrapper for self-post, and we need to set it up. So we need to tell to each of our PG installation where do we have our self-post environment. Okay, our little box of Python with the proper packages. So you click on self-post setup, and here you paste the path that I just told you that you need to have it in hand. So it was this one. In my case, I have it already set up, and I know it works. And then you need to say it's a conda environment. We are not going to use the GPU because we don't have it, and it's version 2.0. And when you have it, you say OK. And please make sure that you don't write anything else but the path, and you don't write any line here behind self-post. Okay, so really the path in the folder. And then to have it set it up, you can go to self-post again, and then advanced. This posting, advanced, and then this one should work. It takes some time because I don't have a GPU. Fine. And then yeah, very good. So this is what you will get. So that is a segmentation, okay, of the image. And then I can try to overlay these segmentations. I will give it some color again as before, like in the morning. And I can try to this one, I think for the overlay, right? But I can try to overlay this mask here. Overlay our image, and then self-post. So we have transparency, and here we have it, okay? And there are some errors like here that we can try to check why it happened. If we go to the original image, if this one is a bit weird, this one, okay, it's overexpressed. But in general, I would say that it works pretty well.
So here, when you arrive to this point, what I would do is to measure whether the accuracy is good for your experiments, or use this to clean a little bit the masks, and there are tools for that. So you can use, for example, so if I say this one, let me just see. And then there's a tool in Fiji that is called I can open this one, it's called Arrate. Okay, so this is a tool to annotate things, basically. So I can say, okay, now I want to open a ladder link. I have it here in my downloads. What's the tip image? I don't remember. Yeah. So then I can see all the ladders here. So this is the one up here. So I can decide which ones are good or not. So I can just zoom in here. Get what is this ladder. So is this 80? Choose the annotation. And just print it a little bit and correct it here. And so on, and get my ground truth data, maybe to return a little bit cell codes if it was not working well, and whatever. Okay, so but I think the segmentation is quite good. And even if you do it with this crappy image here, and then you don't need to annotate all these sort of phase contrast images. I don't know. There are questions? No? Right?
It's hard.
It's hard? So how much time do you spend fine-tuning the protocol in the bench? That is nothing. I want to know the number of hours. Days. Months. Okay, if you spend that amount of months repeating the exercises on this PDF file, and I think you give the class next year. So then that's something that I think is a mistake. So most of the people in the bench, they avoid sitting down to learn programming. They avoid sitting down to learn some technical stuff. And then they never do it, but it takes much less time than fine-tuning any of your protocols. And that's something that I know sometimes it's like a barrier, but maybe think about it in a different way. Yes, it's really worth it for all of you. Even when you need to process some images and you say, I will just do it semi-automatically because I don't want to spend some time writing a loop. A loop for today is many hours from tomorrow. And in a postdoc and so on, maybe it doesn't matter that much, but in a PhD, I think it really makes a difference with your data. And same in structuring your data and your folders, the experiments, the drugs, because then you can automatize almost everything. Yeah. So it's super late. I have again just to leave. So make sure you don't have cables around you. It's a very stupid game that usually I play before, but yeah. Oh, that's fine. Sorry. So no one behind you. Just get a little bit farther from the table. Yeah. Okay. So the game is as follows. Please stand up, all of you. So if you answer correctly, you can sit down. If not, you keep a stand up.
Oh, no.
And if you were sitting and you answer wrongly, then you stand up again. Okay? So, yeah. So the numerical values that we update in a neural network during the learning process are called learning rates or weights. Learning rates 1 hand, weights 2 hands. Up, all. Okay, so you can sit down. So weights are updated after each training iteration step, epoch or epic. So left hand for iteration, right for epoch, and 2 for epic. And so yeah, Florian Cook is a very famous guy in image analysis. The ones who answer wrongly, stand up. Okay, any questions here? Good. Yeah, so if no one was standing up, yeah. So yeah. So a training iteration or epoch or epic has finished after covering the entire training data set. So a training iteration left hand, a training epoch right hand, both epic. You have 2 or okay, so you can sit down if it's the right hand, otherwise it's standing up, if they're standing up. So the number of input images that the network sees before updating its weight is kernel size, layers, or batch size. The number of input images that the network sees, so the number of input images that we use to train the network. So I think you have to stand up, many of you. So what was the kernel size? What was the kernel?
Transformation.
The convolution. So the has nothing to do with the image. The size is the dimension. The layers. The number of filters, right? And the number of times we put it. And the batch size is the amount of images we put on each iteration. Okay. So when a deep learning model is trained with labeled data, ground truth, we are using a supervised approach 1 hand and supervised 2. Supervised. Okay, so you can see that. So, yeah, validation data is used to check for overfitting for the time needed to run one epoch. You are saying one and the other. Okay. Yeah, I think some people need to stand up. Yeah. So we check for overfitting. So validation data is just it's new data. So if does it wrong, is the network is really overfitting to the trained data. So overfitting is common with a small data set. One powerful computational approach is to augment your data or decrease the learning rate. Right? Ladies in the back. You want to Okay, very good. Yeah, but you. The whole, the validation text and the data should be the same. When can even take an image from picture from all of you here. Yeah, so you can sit down, yeah. So no, they are different, okay? So test is the final set that we use to really check if things are working or not, but once we finish the training, okay? And many times they don't, in challenges, they don't give you this data set, so they don't cheat. Okay, so once trained, the model is evaluated using the test data set, which is independent of the training data. Of course. So once the model is trained, it is evaluated with the test data set. I think we agree. Is this test data set independent of the training data? What means independent here? You don't know. So, independent means that the test data set doesn't have same information as the training, right? It's different. Yeah. You need to say something. Okay. Yeah. So it has to be independent. So you cannot cheat in the exam. You really need to. So they need to belong to the same data distribution, but they cannot be dependent. They cannot be the same field of view. They cannot be the same cell, the same organ, the same animal. Okay, it really needs to be independent statistically. Okay. The loss functions used to train the model, for example, cross entropy, mean square error, ensure the biological relevance of the results. True or false? So this, I mean, if I give you a mean square error of 0.031, does it tell you something? Biological. So yeah. So if you really, I mean, these are accuracy metrics and so on, but they are very good for computer vision, but they are not giving you biologically relevant information, right? So if you do tracking, for example, in cells in an embryo development, you want to know whether you are recovering the lineages properly of the cells. You want to know whether the speed is the speed that you recorded in your manually annotated data, whether the activation of the signal or whatever you're measuring is the same. Okay, so it's not the same. Okay, and there's a way to think about it. Imagine you go to the doctor and they give you some treatment and they tell you what the statistical fit of the regression model to check whether this drug testing was working or not is 0.83. And you say, well, are you gonna cure me or not? So, yeah, sorry, I was finding. So, relax and enjoy and hey.